All right, ladies and gentlemen, this is now formally a welcome to the first session on the last day of Savanna Science Network Meeting 2023. And we're going to focus a little bit on large herbivores and how to manage them in a changing world. And then the end of the session, we'll talk a bit about herbivore-plant interactions. Our first speaker, who's getting rigged up here as we speak, is going to focus on approaches to persistence in ecology with some applications to the Savanna system. Can I ask the speakers to just check the program and that you just come in the talk before yours to start to get rigged up by the guys here on the right-hand side. Thanks, guys. Thanks, Sripat. Off you go. Thanks.
Do I have to do something here? Oh, there it is. Okay, so that's my title. Welcome, everybody. And my co-authors here who did all the work are in order: Mark Boyce, Evie Merrill, and Wenyun Zhou. And they have developed different approaches to modeling populations, aspects of population. One of the things that strikes me about the whole conservation and biodiversity movement that is changing the face of interactions with the natural world is that 50 years ago, when I got into this business, we were mostly interested in trying to discover sort of fundamental laws, which we couldn't find. And now we have a new generation of people who are interested in particular objectives. And that is an important area and one in which we can probably make a difference. So I will give you 3 examples of things we can do. Generally, this is the process by which science operates. You start out with observations. You wander around in the field and you get really interested in ants, birds, whatever, and you come back and you say, well, this is really fascinating. How should we think about this? Then you create a model, and a model is just a representation of what's happening out there, which it tells you what you do not want to consider and what you can ignore. And then once you've taken that model, you can formalize it in various ways. And I'll give you 3 examples of how models can be formalized. And once you've got that model, then you go back and you start doing observations based on your representation of the world. And then you get observational data, and then you use some techniques to estimate the parameters of your model. And then you come, in this case, to management conclusions. So the first example I'm going to give you is stuff that Mark Boyce has worked on extensively over the years, resource selection functions. And in some sense, a lot depends on what you are thinking about. So you might be thinking about presence, absence of a species. You might be thinking about abundance. You might be thinking about persistence. You might be thinking about coexistence between species, so you look at presence, absence of pairs of species or guilds or whatever. And the idea is that depending on the species that you're looking at and the criterion, you then look at the habitats in which you are interested, and you assume that abundance or presence, absence, or persistence depends on features of the habitat. And this is the whole process of formalizing a model, where you think about what it is that matters to the things, to the conditions or variables that interest you. So here are examples. You could do abundance or persistence, and then you say, well, this is a function of habitat properties. So you've got vegetation cover, vegetation type, geographical features, and for persistence, for example, you've got human interactions. Now, once you've done that, you can start actually implementing this for specific systems. So this is an example from this paper of the use of these functions, this type of approach, to think about persistence in the western grebe. It's a large bird that's found in lakes in mostly the northern part of the North Americas. Where I am, which is in California, we get Clark's grebe, which looks a lot like that. And this is somewhat similar to the larger grebe that you find here in South Africa. And so this paper was stimulated by the observation that these birds had been disappearing. And so you look at habitat features, which include things like area of water. These things occur on lakes, so area of water, human activities, shoreline vegetation, and so on and so forth. And those are your variables. And then you have the probability of something happening. In this case, it's persistence. In other cases, it might be presence-absence. And so then you do the usual stuff with statistical models that gives you an estimate of the way in which the probability, which is p, depends on each of the individual features. And then you do model selection. You find out what really matters according to some statistical model, which has its own set of assumptions. But we tend to believe most of those, and therefore we proceed. And then you get what really matters. And based on that, you have results that suggest management actions. And these are the two that are pointed out in that paper. They seem fairly straightforward, but one of the purposes of this kind of work is to find out what really matters as opposed to what kinds of good stories we can tell about what should matter. So that's why this approach is extremely useful. The paper also points out that the criteria for persistence are not the same as the criteria for presence-absence. And that's an important aspect of this, which you need to bear in mind. The second example is the work that Evie Merrill has done on how you think about movement of animals. And this is an example taken where you've used GPS movement data on wolves, again in Alberta. And the question is, what do wolves actually do in terms of movement? And this is a fairly common problem with predators and other kinds of animals that you're interested in. Do they just wander about? Do they have a home range in which they wander about? Is there a particular way in which movement is directed or not? And so one of the arguments has been that there are linear features on the landscape. So here's just a picture which illustrates these linear features, and they would include roads, they might include valleys, seismic lines, which are just more or less straight lines cleared for energy exploration. And so what you want to do is you want to build, this is the second class of spatial statistical models, and you want to think about movement in which you use GPS data or other accurate data. And the question that you're thinking about is the contrast between diffusion, which is just sort of wandering about, and advection, which is actually moving with purpose once you find a particular feature. And you can, the original papers on which this work, in which this work was done, include lots of terms like elliptic partial differential equation and so on. And so it sounds much more formidable than it might be. But in truth, it's just a way of representing these two kinds of forms. And statistical methods enable us to take those fancy-looking models and translate them into numerical procedures which tell us that we can build a model in which you just wander around and you don't care about these directed lines, models in which you do, once you find a line, you do stick to it, and you don't wander around too much, and so forth and so on. It turns out that lines do matter a lot, and so once you've done that, then you can go through a model selection procedure, you can find out what really matters and how, and you can then see how spatial structure really matters. And in this case, they found that predator-prey encounter rates are higher in landscapes with higher seismic line density, meaning if you've got these features, wolves tend to use them. And once it's not clear, they don't have a preference necessarily, they might go one way or the other, but they tend to use them, and they tend to do most of their wandering around those lines. And as a result, if there are prey, whether they're at low density or not, they tend to find these prey if they're near the seismic lines. And standard predator-prey theory, of course, assumes that prey density matters, and that the higher prey density leads to a higher likelihood that predators will find them, and that's obviously not good for the prey. But in fact, in this case, when you have these features, you get good encounter rates at low densities. So prey at low densities can be at risk in these kinds of habitats unless they learn how to avoid these sorts of linear features in the landscape. And of course, we all know a great deal about this kind of thing. In the parks over here, you do a lot of wandering around on the roads, and a lot of animals are attracted to the roads. So that's the sort of thing that you can make use of. And the final class of models that I'm going to talk about are somewhat more abstracted in representational thinking, and they are analytically based models in which you contrast species characteristics rather than landscape characteristics. So it's well known that you can take data on species over time, and you take a species, you take individuals of the species, you have repeated measures, and you can estimate what I call vital rates. And by vital rates, you mean things like growth, survival, reproduction, and so forth. And once you do that, you can estimate these rates. But these rates are averages. That is, you've got 25 individuals, 10 of them actually fall in a particular year, and so you estimate a fertility rate and so on. But fertility and survival are random events. That is, we don't actually know that a particular individual is going to die or is going to give birth or is going to successfully raise offspring and so on and so forth. All that you can do is you can say, well, this is what we expect. But since they are random events, the lifetime reproduction, including survival, which I call LRS, it's also a random variable. That is, some individuals just don't do anything in terms of reproduction because they die young. And we know a lot of examples of that. So I'll give you an example. This is the European version of the data for red deer, which is a form of elk. The models were developed by Jean-Michel Gaillard. And on average, this population that he studied and for which he made these estimates kind of replaces itself. So typically, one female is giving birth to one female. You think, well, some females give birth to somewhat more offspring, some to somewhat fewer. You got yourself a nice bell-shaped curve sitting around one. But then we figured out how to compute these actual distributions of this random variable, and this is the sort of thing you get. There's a lot of individuals whose probability of having 0 offspring is high. And then this arrow is the mean, which is totally unrepresentative, in my view, of the distribution. And there are a substantial percentage of individuals doing a lot better than you would think. So, as a result, the distribution of lifetime reproduction is highly skewed. And this is true for every species that we've looked at. Why do we care about this? One, you might say, well, we care in a general sense, but in a specific sense, the reason that you care is that small populations go extinct. The extinction probability of those populations depends on the distribution of their lifetime reproduction. If most of it is individuals doing a lot of offspring, then you don't care. And so the distribution matters, and we can compute this. So it turns out that the distribution is important, unimportant really, for that kind of species, and examples are trees, shrubs, and so forth, salmon, a lot of fish, and the distribution does matter a lot for mammals. So that's important, and the reason the distribution matters when things are a little better for the offspring is that the way in which you live really matters. You can increase the survival rate of offspring, you can produce slightly larger numbers of offspring, you can do it later in life, and so on. That's the final message. Thank you.
Thank you very much, Sripat. You timed it very, very well there for us. I'm sure that there's some useful discussion. There's a lot of modelers in the room, and I think they will definitely enjoy talking to you over tea time. So thank you very much. All right, we're going to move on to assessing the potential of large herbivore regime shifts, specifically under climate change. So Reese, please entertain us.
Okay, hello everybody. So I'm Reese Thornley. I'm a PhD candidate at the University of Aarhus, and today I'm going to be presenting my second PhD chapter, which is something I've been working on a lot with Kate Parr. So we're essentially looking at how large herbivores are going to respond to global change within savanna protected areas. And so really we need to start thinking about, what protected areas are, and as you know, these are one of the main ways in which we've tried to conserve megafauna over the last sort of 50 to 70 years. And we're lucky because we're in actually one of the best examples of a well-funded and well-managed protected area. But if we look on a cross-continental level, if you pay attention to the red parts of the pie charts, what you'll see is actually that overall protected areas are severely underperforming. And what this means is that they're failing to fulfill their key mandate to protect and to conserve megafauna. And so as a result, megafauna have actually been lost. So large herbivore populations have declined over the last 70 years within many of Africa's protected areas. And this is often as a result of things like human-wildlife conflict, competing land uses with livestock, cropland, that sort of thing, and also through the pervasive illegal harvesting of megafauna for things like bushmeat and the illegal wildlife trade. And so protected areas in Africa are really representative of this broader biodiversity crisis in which the world finds itself. And so the wildlife within these protected areas is really in a lot of trouble. But one thing that we need to remember is that in the background of the biodiversity crisis, there's been another crisis which has been unfolding over the last 70 years, which it comes from the exponential growth in anthropogenic CO2 emissions and greenhouse gas emissions. And this is of significant concern because this is going to result in, already resulting in the complete restructuring and the complete change of the Earth's biotic and abiotic conditions with really pervasive impacts for the biosphere. And what we're seeing is more and more extreme conditions, even now, with some of the hottest years ever recorded all occurring in the last 20 years. And so there's only one way to describe this absolute mess and this lack of inaction that has caused this problem, and that is, it is basically a crisis. There's only one way to describe this, and we are in the climate crisis. And so what I'm interested in is how this is going to affect protected areas, because we're already seeing the impacts of climate change manifest within protected areas. And the only thing that's certain about the future for protected areas is that the future will change. And the relative climatic stability that protected areas have enjoyed over the last 50 to 70 years is a thing of the past. We need to forget about climatic stability, and we need to start to embrace the fact that the only thing that's certain about the future is climatic instability. And we're likely to exceed our climatic safe space. So according to the Paris Accord, 1.5 degrees of warming is our climatic safe space, but we're currently on track to exceed that safe space within the next 20 years. So we need to start thinking more long-term about how this is going to manifest to affect protected areas and affect large herbivores within these areas. Because what we're going to experience within protected areas is an unprecedented change to the biotic and abiotic conditions. And it could be to something that is completely unrecognizable from what we've seen over the last 70 years. And it's going to be at a rate and pace which is unprecedented in human history. And so what I really want to know is how this is going to manifest. Is this going to erode the conservation capacity of protected areas in the long term? And so what we really want to know is what are the consequences going to be? And we need to start planning for the future. And so understanding the consequences now will give us more capacity to adapt. And if we think about the future, we're currently on track for 8.5, so RCP 8.5, so that's basically an emissions pathway that is equivalent to 1,200 parts per million of carbon and between 4 to 8 degrees of warming, which is basically sounds like Armageddon. So that's the current trajectory. RCP 2.6 is the Paris Climate Accord. Basically, forget about that, because unless we transform our society in the next 6 years, it's not going to happen. And so one of the only really likely other scenarios is RCP 4.5. The RCP 4.5 basically is 600 parts per million of carbon, and between 2 to 4 degrees of warming. And so this is a significant divergence from the climatic conditions which protected areas have been subject to over the last couple of millennia. So my aims, so I want to know how the climate crisis is going to alter the abundance of large herbivores by 2080 to 2100, and if this impact will vary spatially. So we know that Africa is a very heterogeneous continent, different jet streams and that sort of thing, and so we can expect different impacts between the different regions. Okay, it's supposed to come up one at a time. Anyway, spoilers. So basically, to estimate this impact, what I first did was I needed to know how many herbivores there are, where they are, what is the abundance of herbivores within protected areas, and to get this information, I collected aerial survey data from across the African continent for many African savanna protected areas, and this is basically equivalent of 333 survey areas and 750,000 kilometres of protected African savanna. And then we then needed to think about how do we model this, what kind of variables do we include. So we included 7 natural variables. So this is like rainfall, water availability. And we also included human predictive variables. And we're basically, by doing that, we're accounting for the fact that humans are having, in many places, a disproportionate influence on megafauna biomass. And so we're basically accounting for the fact that climatic variables in some areas may not necessarily be the thing that is driving the densities. And then, we use boosted regression trees, and then what this gives us is it basically estimates the importance of the individual predictive variables. And so what we expect is where the predictive variables have like a low climatic predictive variables have a lower importance, we would expect the influence of climate change potentially to be lower. Where the relative influence is higher, we would expect climate change to have a higher influence. And so basically what we did is we went to the IPCC report, looked at the downscaled data, and basically changed the baseline data. So the data that we used for 2015, we then changed the predictive variables to match the future scenarios for RCP 4.5 and RCP 8.5. So we changed rainfall, and we expected to get more declines in southern and central Africa and increases in East Africa, with more severe declines, more significant increases with RCP 8.5 than 4.5. And so it was similar for surface water availability. We used seasonal and permanent water for that one, and then also vegetation seasonality, which we expect to increase in southern and central Africa and decline in East Africa. And then we used woody encroachment because we know because of increases in CO2 that we expect the spread of woody plants basically into the savanna biome, which represents a structural change to the savanna canopy. So we also included that, which is going to increase in both scenarios. And what this gave us then was six predictions per scenario. So we have for elephants, and then for the 5 different functional types, which was created by Gareth, and I think by Sally somewhere as well. So thank you for doing that. That's made it a lot easier for me. So then we ended up with basically 6 different predictions per climate scenario. Now we get to maybe the interesting part. There's enough context, so we'll go on to the good stuff. Okay, so if you look, I'll just explain the graph to you quickly. So there's these dashed lines that represents the no change scenario. And anything below the line basically represents a decrease in biomass, and anything above represents an increase. And what you need to note is that for the different functional types, they have different body sizes, so the y-axes have different values. But if you look at this, what you see is that there's very clear losers and very clear winners on a cross-continental scale. So the small guys, so the dikers, those sorts of things, they're going to love it. It's going to be, they're going to have a great time. But then if we move over to things like elephants, non-ruminants, grazers, what we see really on a cross-continental scale is quite significant declines on a continental scale, which means that protected areas, in terms of the composition and the abundance of herbivores, is really going to be transformed. And if we think about the second aim was to look at how it varies spatially. So we can break this down by the different regions of Africa, and what we see as a consistent pattern is more severe declines in southern Africa and central Africa compared to East Africa, with many of the declines being much more significant, much more severe than any of the increases. And so some of these declines for elephants, for example, represent 8,000 kilograms, and that can be, and that's in places in Botswana, that represents between a 70 and 80% decline in the elephant populations. And some places like Etosha National Park, that's going to represent a complete extirpation under RCP 8.5. So there's going to be some significant consequences. And what we see now, and then what we see basically is the small guys start to be the winners, and the browsers, although there is a tendency to decline, the decline represents a much smaller proportion of the total population. And so there's basically going to be this, this gradual progression towards more water independent and more browsing species. And if we look at this, what it looks like spatially, you can see under 4.5 that many of the declines are concentrated in the Okavango Delta, with much more significant declines here, under 8.5. So this represents a really, really huge shift in the ecological capacity of these protected areas, which is really worrying. And you'll see that some of these protected areas, like Selous in Tanzania, they're going to be the places that increase. So these increases in rainfall are going to be things that drive higher megafauna biomass there. But overall, we're looking at much more significant declines than increases. Now for megafauna, for all the other functional types, it's a much more of a mixed picture, so it's not as much of a clean story. But what we do see is that a lot of the significant declines are concentrated in the Okavango Delta, and with much of the really significant increases being in East Africa. And so what this actually could represent is basically community downsizing. So one of the consequences of the climate crisis could be this shift towards smaller bodied, water independent browsing species. And so what we'll end up with is a situation basically where we could end up with feedback loops. So it could be where we have community downsizing towards these smaller species, reduced top-down regulation of ecological processes, savanna structure, savanna heterogeneity, which then, when you combine that with increases in CO2 and increases in rainfall in East Africa, these are all conditions that promote savanna degradation through woody encroachment. So what we're going to see is a change in that large herbivore community with huge implications for savanna structure and savanna persistence in the future. And what that's basically going to do is that's going to severely erode the conservation capacity of protected areas. And so there's a couple of key points. There's going to be this huge reduced ecological capacity in many central and southern African protected areas. And what this enables us to do is form conservation priorities. So the areas with the most significant declines are the areas where we really need to start adapting and mitigating these consequences. And the two biggest drivers of declines are going to be woody encroachment and reduced permanent water, because all megafauna rely on access to water to some degree. And if we lose that in many protected areas, you're basically going to lose a lot of the wildlife. And I want to end by making just one point. So this is a different challenge to the challenges that we've experienced over the last 70 years, because we have very little agency over the degree of change that we will experience. This is something that's being forced on protected areas. It's being forced on African nations, and African nations have contributed relatively little to the climate crisis. But the key thing here is that we need to start preparing for the future. So we need to start to adapt so we can overcome, so we can mitigate. And the first step towards adaptation and mitigation is trying to understand what the impacts will be. And I hope that this research has contributed to that in some way, so we can maintain the conservation effectiveness of protected areas in Africa. So thank you very much.
Thank you, Reef. You've done well. We have got time for one quick question. Thanks, Sharon.
Thanks, Reese, for a great presentation. I was wondering if you included any interaction effects between the species. Thinking in terms of elephants, if the rainfall decreases and there is a resource depletion in an area, they would have a knock-on effect to species that they're living in.
Yeah, so no, so there is, so there's the model, the model doesn't work like that. So the model's like individual species, and it doesn't account for interactions and knock-on effects between species. But there is interactions between the predictor variables, but with the kind of modeling framework we use, that wasn't possible. But it's something that I have thought about, but I'm not really sure how to do it mathematically, basically. But it would be cool to do that if anybody has any ideas.
Thank you, Reese. I'm sure there could be some discussions afterwards. All right, yesterday Chloe was moaning about a large elephant that was causing trouble in this room. Now Chloe, Tim is going to help you to understand why elephants are large. Thanks, Tim.
Thank you, everyone. So I've been coming to this conference now for several years, and I've heard some really fantastic talks on elephants. But the elephant in the room has always been quite, why are they so big? And so I've often asked people, I said, why are elephants so big? And I've heard various answers. So some people have said, it's because they don't want to be eaten by lions. Well, neither do I, and I'm not the size of an elephant. Although given the amount I've eaten and drunk this last few days, it's possible I will be next year. People have also said it's because they need to eat large amounts of low-quality food. And again, I don't buy that, because that's the wrong way round. They need to eat large amounts of low-quality food because they're big, but it's not a reason they got big. There are many small animals that feed on similar things. And so I thought I'd try and think a little bit about why animals get big in general, and focusing on elephants. And so if you look at the theory that's out there already on body size and life history, most of it says that you should kind of live fast and die young. And the reason for that is if you're able to breed at an earlier age, and therefore a smaller size, than your competitors, you're going to get your genes out there more quickly. It's a little bit more nuanced than that, but that's kind of really the basis of it. And so a lot of theory out there essentially says, well, you should live fast and die young and not grow to a very large size. And of course, there's lots of small animals, insects, various reptiles, amphibians, and mammals and birds that do that. And so the existing theory largely says that elephants should be impossible. But of course, not all elephants are big. In the fossil record, we have quite small elephants and small rhinos and small hippos on islands. So when elephants have got stuck back in evolutionary time on oceanic islands, and the sea has increased in size, particularly in the Mediterranean, you saw evolution of large species to small species. So elephants don't need to be big. Historically, and from the fossil record, we know they can be small. And that's part of something called the island rule, and in fact humans also adhere to the island rule, with evidence of Hobbit man or Homo floresiensis on the island of Flores. So elephants don't need to be big. There are small elephants, or there have been small elephants as well. And of course, there are lots of other animals that are big as well. There are things like T-Rex. That didn't eat much grass, I can tell you. There are also things like rhinos and whales, and also some of the large bears that are omnivores. So you can get big, not just by eating grass and vegetation. You can get big by eating meat, and you can get big with an omnivorous diet. And so what we really want to do is perhaps try and come up, and I think this is kind of what theory can contribute. We want to try and come up with some general understandings of what it is that drives animals to large size, when most of existing theory says that they should be small. And so I'm going to talk you through a really simple model. It's a very simple model. There's a lot of complexity that's not in there. So, feel free to put your hand up at the end and say, well, why didn't you put this in? Why didn't you put that in? The reason I didn't put them in is I wanted to have a reasonably general model that would allow me to come up with some insights into the general processes that allow animals to grow large, and not just elephants. However, I would love to parameterize this for elephants, so any of you elephant researchers out there, if you're interested in this and you have some data, come and talk to me and we can perhaps collaborate to do it. So my model has two age classes, juveniles and adults. And the juveniles don't reproduce, so they're just growing to get larger. And then when they become adults, they reproduce. I'm going to make the model density dependent, so the population will grow and remain about the same size. Well, it will remain the same size over time in this model. And I do that because, on average, over evolutionary time, animal populations that persist are fluctuating around an average size, and that average size doesn't change very much. The average size is constant, and that's why we're not knee-deep in things like elephants. And I'm going to vary age and size at sexual maturity and the growth rates of my fictitious animals in my model to see what happens. So this is a model built on what Tulja referred to as vital rates earlier. And the way I'm going to do it is I'm going to compete various different strategies against one another. And so down here, these are the growth trajectories. So this is age, and this is how big you can get. Don't worry about the units. This is just kind of the numerics that I did. And so down here, we have a strategy that grows and starts breeding at a small size and a young age, and then it asymptotes. So these are things that show determinate growth. And then up here, we have something that grows for much, much longer to a larger age, and it starts reproducing at that dot there at a larger body size. And again, it continues to show deterministic growth. And based on a rule that's kind of approximately correct for mammals, we have individuals reaching sexual maturity at about 80% of their asymptotic body size. And then so I said, well, let's think about how body size and survival might vary. And so for all of my strategies, I'm going to compete against one another, and these are the sizes at sexual maturity. What you can see is these individuals reach sexual maturity at a young size, and they've got a relatively low survival rate. But if you grow for longer, and of course it takes you time to grow for longer, as you saw on the previous slide, you have a higher survival once you reach adulthood. Density dependence, I put density dependence in reproduction, and so when density goes up, reproduction goes down. And what do we find with this very, very simple model? So individuals don't breed until they reach sexual maturity. When they do breed, they produce offspring in a density-dependent manner, and we've got the same survival function. Well, what's really interesting is we find disruptive selection on body size. So this is the point of minimum fitness. We can show in our models, we can show mathematically that carrying capacity is fitness. And so below this point, you select for small elephants. Evolution drives you in this way for small size at sexual maturity. But above it, it pushes you in the other direction. And so what's going on? Well, so there's two things that are happening. So first of all, if we look at the proportion of individuals that are born that survive to sexual maturity, we find this curves downwards. So we can see it curves down, and that dotted line is the point of minimal fitness. Whilst over here, this is life expectancy, so how long you're going to live for once you've reached sexual maturity. And this curves upwards. And so these are the two key components of fitness in this model, and they combine to give us that shape there, that disruptive selection. So what's going on in this model? So in this model, we know what fitness is, so we can work out who's going to be the fittest strategy. And reproductive failure, so your failure to breed, is caused by density dependence. So the strategy that wins is the one that can live at the highest density, or the highest biomass works as well. And so body size trajectories can counter the negative effects of density in this model. And so we have an evolutionary stable strategy where you trade off the mortality rate between birth and sexual maturity with how much reproduction you're going to do after sexual maturity, which is a function of how long you live for. So a strategy can carry costs of quite high juvenile mortality as long as, when you make it, those few individuals that make it to adulthood live for a long time. And so what that means is something like an elephant is going to must have, to get large, you have to be able to have high life expectancy at sexual maturity. You must be able to live for a long time. You have extended growth rate. So in other words, you're a juvenile for a long time, and those two things trade off. And I suspect that what happens in many large animals is that predation on the juvenile stage is kind of what's key here. And so selection for large size is probably, in general for large animals, is generally a way to escape a cause of mortality that happens early in life. But there are other things that can kick in that can help you deal with that mortality pressure, including things like extended parental care. And so the next step is to try and make these models a little bit more realistic. And one thing, to bring in things like senescence. So of course, not all animals are small and not all animals are big. There are some in the middle. And so we need to work out how you end up with an average-sized animal like an impala. So maybe next time I'll give a talk why are impala sort of the size they are. And we also need to bring in some stochasticity, so some environmental variation. But what I think we've got I think we've now got a simple model that shows the trade-offs in fitness across the life cycle can align in such a way that to kind of overrule the findings of previous theories that everything must be small. There is parameter space when you can grow to be big, and that's as long as you grow to a size where causes of mortality on the landscape are relatively unusual. So you've got to a size where not a lot kills you. Interestingly, a consequence of that would be that if you start hunting elephants, or continue hunting elephants, adult elephants, you increase their mortality rate and you would eventually end up selecting for lower size. So lots of people to thank. This was a paper we published last year in Peer Community Journal. It's free to submit to, it's free to publish in, and you get an impact factor and everything. Okay, and thanks to all of the co-authors on that paper. And, oh dear, some time for questions.
Thank you, Tim. Indeed. And some of the smaller people in the room is asking questions now.
Thank you, Tim. Such a nice talk. I have a curious question. So in your model, you said there's adult and juvenile, the elephant, large elephant, small elephant, and adult and juvenile. What is the mortality rate for a juvenile of an adult, a juvenile of a large elephant compared to an adult of the small elephant if they are the same body size?
That's a very good question. I do assume that survival changes with body size, so a small elephant will have a lower annual survival rate than a large elephant, but that's why I'd love to collaborate with people who've been studying with elephants who will be able to use their data, parameterize my model for elephants, and my model will almost certainly be wrong. All models are wrong, and I'll probably end up concluding that all elephants should be small.
Right, we're going to take one more question. Mark?
So one thing that didn't seem to make sense along the way was your comments about carrying capacity. And you can have a lot more animals, small animals, than you can large ones on the same resource. And then the second comment, you made a final remark about stochasticity. Seems to me what you're missing is seasonality, because that's what's going to drive evolution of larger size for mammals.
So first of all, let's deal with the stochasticity first. Yeah, I want to put that in, and I think it will be interesting to see that if we have fluctuating seasonal mortality rates, whether the average of those are sufficient to drive the patterns that we get. It may be entirely possible. Second, I didn't have time in this 15 minutes, but we actually go through and we can show you get the same results when you work with total biomass as fitness rather than carrying capacity. So you multiply the size distribution of animals by their relative abundances, and the results hold. But I didn't have time to go into that. It works for the model.
Would like you to respect the chairperson here, please. I may be small, but still relative. Please carry on with the discussion afterwards. I think it's very important. Thank you very much. Thank you, Tim. All right, we've got a short little rush now, a little 5-minute talk by Darren on the legacy effects of one of these big things die.
Thanks, Sam. So I'm going to give a little 5-minute teaser talk on a project that's just starting to understand the ecological legacies of Tims and possibly big elephants. And this is a big collaboration amongst all the different entities you see at the top, and I'm really fortunate to work with a bunch of amazing people and scientists here. So when we think about mega-organisms, we know mega-organisms across lots of different ecosystems have really long-lasting ecological legacies on ecosystem processes. If we think about great whales in the ocean, they move nutrients from the poles to the tropics, from the seafloor to the surface. They're really important ecosystem engineers while they're alive. But when they die, their carcasses fall to the deep ocean, literally transferring tons of carbon and nitrogen to essentially this deep sea desert. These whale carcasses act like hotspots of biodiversity and ecosystem processes that last for decades, and we call them whale falls. We know that mega herbivores and terrestrial systems are really important ecosystem engineers, but what happens to their carcasses when they die? Do they create these similar ecological legacies in terrestrial systems? So we're essentially asking the question, are mega carcasses, elephant carcasses, the whale falls of African savannas? And what's really important is that a 4,000 kilogram elephant is about 800 kilograms of carbon, 300 kilograms of nitrogen, and 120-ish kilograms of phosphorus. And that's deposited to the savanna landscape when they die. A lot of those nutrients get eaten by scavengers, but a lot of those nutrients also make it into the soil. And this cartoon kind of encapsulates our hypotheses of what happens at a megacarcass. That initially we get a lot of carbon and nitrogen into the soil, right, that activates the microbes, increases soil respiration, changes microbial biomass and diversity, right, and they chow through a lot of those nutrients relatively early on, especially things like ammonium that are initially toxic to plants. After a few years, right, we should get plants recolonizing these elephant carcasses sites, right, increasing plant diversity, potentially creating unique hotspots of diversity. Right, and after a couple of years, the bones start to leach phosphorus as they age. And as this stoichiometry of nitrogen to phosphorus begins to change in the soil, it should facilitate plant growth, potentially facilitate herbivores coming into these sites to come eat the nutrient-rich forage. And we estimate that these megacarcass effects last potentially up to a decade. So there's about 300 elephant carcasses in Kruger every year just based on the populations of elephants and their mortality rates. And if these carcasses or these carcass effects last for up to a decade, then at any one time there's about 3,000 elephant carcass hotspots impacting the savanna landscape. So how are we testing these hypotheses? So we're choosing 20 elephant carcasses of various ages in both the granitic and the basaltic soils to test for the impact of initial soil fertility on the role of these megacarcasses. We're centering these 15-meter radius circles on the center of the impact of the carcass, and then we're measuring a variety of different metrics radiating out from the center of that carcass so that we can understand essentially the influence of what we call the carcass sphere on community processes and ecosystem dynamics. Right, we're looking at a bunch of different metrics in the soil about nutrients and microbes, production and plant diversity and how these nutrients actually move out into the plants, and how these carcasses affect the abundance of vertebrate and invertebrate herbivores and how they affect plant communities. Right, so we're really at the stage now of identifying our carcass sites using the amazing database that Kruger has on elephant carcasses. This is one of the carcasses we found up near Satara a few months ago. It was a big bull that died just south of the Swanee River. You can see the big impact area of the elephant carcass. You can see our transects radiating outward. This is a carcass that was about 4 years old, just southwest of Skakusa. You can see most of the bones are still kind of in the center of the carcass sphere, right, and the big, large area of smaller grasses surrounded by this area of taller grasses. But we know that not all carcasses are alike. This is a carcass that was just 2 months old. You can see there's no bones anywhere. So one of our emerging ideas at the number of scavengers is really important for mediating probably the size and the duration of these carcass spheres and the legacies that these elephant carcasses have on African savannas. And this project is just emerging. We're continuing to build collaborations. So if this little teaser talk kind of piques some ideas in any of y'all about how you could potentially join our project and collaborate with us to kind of unlock the mysteries of elephant carcass legacies, please come and chat to me. Thanks.
Fabulous job there, Darren. And I see Urs has got a question, and he's going to chat you at tea time about that. And it is a nice teaser talk there. So we're going to move on, and we're going to have Lorenzo predicting for us where rhinos is going to be, certainly at a smaller scale that may help rangers to protect them. Thanks, Lorenzo.
Hello. Okay, so my name is Lorenzo Ruaro. I'm part of the magic team of Stellenbosch University's mathematicians that do or try to do ecological stuff. And I'm starting to say that the title might be a little bit misleading. So I'm not really focusing on the space-for-time substitution, but rather should be called the spatio-temporal synchrony of population dynamics of white rhino. So let's start off with a little bit of introduction. Most of you might know that white rhinos are the numbers are dwelling in Kruger National Park, as they are labeled as near threatened by the IUCN. This graph is a little bit old, but for the purposes of our project, it's good enough. So our project would like to focus on the drought effects on white rhinos population and how it affects the mortality rates of white rhinos. It was particularly, especially the 2015-2016 drought was particularly harsh on the white rhino population since they are grazers. And so we would like to see how the environmental variables fluctuations affect the population dynamics and the mortality rate of white rhinos. So the project uses helicopter sensors, which are good enough for our project because we will likely not see the effects of poaching since it peaked in 2010. And of course, due to the nature of the sensitivity of the data of the rhinos, I cannot show you spatially where they are. So we took Kruger National Park, we divided it into 5 kilometers by 5 kilometers square cells, and then we counted the individuals in each cell. And so we took into account the population for a certain cell I at year t, and we calculate the relative growth rate for each cell at year t. Just to make a small note, a mathematical note, if the population either at NIT plus 1 or NIT were to be 0, we would discard that year because we would have problem calculating the relative growth rate. Just to include the fact that this relative growth rate takes into account immigration and emigration rates from one cell to the other, as well as recruitment rates. So, what is spatial and temporal synchrony of populations? So, what we want to look at is how each cell influences the other through space and through time, and we do that by calculating the correlation between the population and the relative growth rate of each cell. In this example, we can see population A that is very close, spatially close to population B, and their time series compared are quite similar. And we can see also that in the correlation plot, since this pairwise coupling is close to 1, which would mean perfect synchronization. And we can see, again, in this example, that usually correlation decays over distance. And it reaches 0, which means basically independence of the different cells. If it's -1, we have a complete negative synchronization, and that usually is the case of predator-prey populations. Anyway, so starting off with some preliminary results, so please don't bash me if I'm trying to interpret them. I won't say, for the people who were present at the bioinformatics symposium, I won't say rhinos move randomly. So what we have here is we took a snapshot of Kruger National Park, and we compared the cells to each other over distance. So we have on the x-axis our distance that goes up to 80 kilometers, and on the y-axis we have the covariance, which is, for the sake of the simplicity, is our correlation in this case. We are not taking into account the covariance of the populations of each cell because it wasn't significant, but we can actually take a look at how the relative growth rate of a single cell is correlated to the relative growth rate of another cell that is spatially distant from it, as well as the population size and the relative growth rate of a different cell. We can see both in the covariance graph of the relative growth rates and the population and relative growth rates that we have oscillations. And this, from a spatial point of view, might indicate the presence of preferred areas. So if we have, for example, a high population until 20 kilometers, the relative growth rate of nearby cell would be negatively, 10 kilometers, would be negatively affected by it, and then it will have a growth which might indicate, of course, the presence of preferred areas. The last spike might be a saturation effect due to the fact that we reach the width of the park. And so, yeah, this is important to take into account as the cell size and the scale of which these simulations are taken, these observations are taken from, might be relevant. Going to the time synchronization. So in this case, we are taking into account a single cell, and we look at the single cell throughout time. And in this case, the covariance of the relative growth rate versus the time, which is the x-axis, the time window is only 20 years, is not significant. However, as for the spatial correlation, we can see that the covariance of the population is consistent with the covariance of the relative growth rate and the population dynamics. And what we can see here, what we can actually try to infer from this graph is that basically we have an oscillation most likely due to depletion of resources and to oscillation in the environmental variables. But what is most interesting is this fall at the end of the graph, so like 20 years. So in this case, we can see that if we had a cell with a very high population, it will go, the relative growth rate after 20 years would dwell. And this might indicate either the presence of a very harsh drought or the start of the poaching pressure. So that were the preliminary results. So the next steps are basically combining the spatial and temporal covariance into a 2D heat map to infer how the two are correlated and develop a demographic accumulation curve, which ideally is very similar to the species accumulation curve, where we have here, in this example, the mean of the relative growth rate of and the demographic accumulation curve. That is how the mean of the relative growth rate changes over the number of sites we are looking at, so the cells, and as well the standard deviation. Also, we would like to incorporate the Moran effect, which is how the oscillations of environmental variables, and in this case we would like to look at the rainfall and temperature, affect the oscillation of population dynamics, as well as looking at the sensitivity of the results with respect of the cell size, so how the cell size affect those curves. And of course, we would like to include other censuses, as the helicopter one is taken only in winter months every year, so we would like to incorporate this seasonal aspect. And last but not least, to go back to the title of my presentation, we would like to validate the feasibility of using a space-over-time substitution approach, which would mean we take a look at a single cell, we assess the synchronicity levels of the cells around it, and we try to infer the temporal dynamics of that cell, looking at the spatial correlation of the spatial correlated cells around it. So, many thanks to my collaborators and South Parks for the data, and of course, last but not least, Oliver Schurmans. I hope I didn't butcher your surname, but thanks to you, because he was an honor student under my team, and I used this code to basically get these results. Thank you very much.
Thank you, Lorenzo. You've done a great job. You've left a lot of time for a bit of questions out here. So, any clarifications, questions for Lorenzo? Oh, no, you convinced him. I don't know what.
Yeah, so.
Perfect. You know. Yeah, we've got him. He's woken up.
So when you were thinking about the autocorrelation with respect to time, how does the generation time factor into this? I mean,
I would have liked to answer you, but the census we have do not take into account age classes. So yeah, otherwise that would have been better. But yeah.
Are there any other questions for Lorenzo? Maybe I can reflect on one or two things for you, Lorenzo. We have noticed there's obviously been a substantial decline in rhino numbers. In the core areas where they have retained those identifications that you've highlighted, we've had a substantial decline, but in the old areas that was good places for rhinos that were killed out, we've had an incredible colonization rate. So I think some of those spikes that you're seeing at the end there are some of these movements that rhinos are busy making in response to lots of suitable resources, etc., that's happening across the landscape. Okay. You're happy with that? Cool. All right. Actually, Adrian did negotiate with me. You know, this is how you do it in South Africa. You know, you have a little bit of a deal behind, etc. But luckily, he must have negotiated with Lorenzo as well, so he didn't put me in a difficult space. Adrian has got a 5-minute talk. He tells me it's 10 seconds over. So, Adrian, you've got a bit of leeway here. Thanks.
Right, okay, cool. Since I've got a couple of seconds, I was going to change the title of my talk to fit in with the general aspect of the conference and make it termites on fire and other fun kids experiments. But let's stick with what I know, so I'm going to talk about rhinos. So one of the things that's happened, all of you are well aware that we are facing a rhino poaching crisis from about 2008. And in response to that, we've seen privates and provincial parks go through a large number of anti-poaching methods. And one of the most prominent of this has been dehorning. And it makes sense, you know, go along, cut the horn off, reduce the thing that poachers want, and increase the probability that an individual will survive. But there's been concerns raised on this, and it has to deal with the potential behavior and welfare problems that come about removing this important aspect of the rhino. So in response to this, there's been studies looking at intercalving intervals, changes in calf survivals, changes in hormone changes, both before and after dehorning. But one of the things that hasn't really been looked at is spatial changes. Now, I say that knowing full well that Vanessa was up here last year talking about changes in black rhinos, but we know that black rhinos and white rhinos have very different social systems, and a large number, more white rhinos are being dehorned than black rhinos. So it's vital we understand what's going on there. So to this, we put together very simple objectives. We wanted to determine whether white rhinos alter their territories, and there I'm talking about adult males, and their home ranges. This is adult females, and then subadults of male and females after dehorning. We were interested whether there was a sex difference, which it varied between males and females, and also an age difference, because adults are more established on the landscape than subadults that are still moving and trying to find where they are. So we did all this work at the Atala Game Reserve. The reason we worked there is that all the rhinos were dehorned. All of them are well known. They all have notches, and regular sightings of them are recorded. This allowed us then to generate home ranges, and we did this 2 years before and 2 years after dehorning. And the reason we use 2 years is because at 2 years they have to dehorn them again. So we're dealing with the first incidence of dehorning. We limited the individuals in our study to any individual that we could get 20 independent samples before and 20 independent samples after, so the sighting data there. This gave us sample size of 15 individuals. This is 45% of the population there. Adults, this is 4 males, 5 females, and subadults, it's 6 subadults, 4 males, 2 females. So what did we find? Well, we found an age and sex interaction. That was driving it, and it's being driven by adult males. So what we saw is that there was a significant change in the size and location of the territories of these adult males. When we talk about size, these territories contracted. They contracted between 18 and 36%. When it takes the size, we had 2 individuals. I had one individual I could generate home ranges for, okay? The other one we had the dots, but they actually packed their bags and moved from the eastern side of the park to the west. And the one that we could generate the territories for, the closest of those edges of those territories was 6 kilometers. So the core of this moved quite far. The other one we had points, and again, they were on that side of the park before, and then afterwards they were on that side of the park. So what does this mean? Well, the key thing about male rhinos is that they use their horn and they defend mutually exclusive territories. Okay? So is this a situation where we're seeing horn loss is reducing their ability to defend these areas? Right? So if we take a look at size, it could be a situation that the proportions that they've lost, why it's shrinking, is that they've lost these 2 neighbors. Okay? It's possible. We're looking at the data, and that doesn't seem to really be the case. But what we then do tend to find, this has been shown in a number of species, that as soon as an individual feels threatened, it reduces the area that it actually uses. So it shrinks the area that it actually has, okay, and will utilize. So what we're seeing here is it could be that males are using a smaller portion of their territory, getting away from surrounding males, reducing contact, and thus reducing the possibility of losing that. The large range shifts, this could be a situation where it got chased out. Maybe a male is young, or it has a long horn, it's very good at using it, it loses its horn, now everyone's on the same length, the next door neighbor or one that moves in is bigger and they're able to take over. But we don't have those kind of behavioral observations to back that up. So what are the long-term aspects of this? Well, the key thing is that we have to understand that the changing of territory ownership is a natural process. We didn't see chaos. It wasn't that every single individual and all the males started shifting their territories all over. Okay? We saw a few, more in kind of a natural. The other thing is the shrinking of their territories. Territories sit within larger female home ranges. So it wasn't that it stopped interactions between males and females, all right? So breeding opportunities are still there. The one thing we don't know is how these males, the extent to which females use horn size as a sexual selection, and so these males that have moved from one side of the park to the other, what their potential short-term and long-term breeding opportunities are. Okay, thanks very much.
Thank you, Adrian. We certainly have got time for a question or two to Adrian. Any questions?
Oh, there we go, Lisa.
Thanks, Lisa. She's chasing me on, eh?
Thank you, that's really interesting. I just wanted to know, and maybe I missed it in the beginning, but so to what extent, how do you know that these shifts and making it smaller or shifting it entirely is a response to not having a horn instead of a response to just having been manhandled and like the trauma of the dehorning?
Yeah, well, that's a good, yeah, how do you tease those two apart? I mean, no one dehorns without manhandling and darting the rhinos, right? So we don't have individuals that have lost it. So yeah, it's maybe that whole process. The one studies looked at the actual stress involved of it, and you find that their stress peaks after being darted and dehorned, and then it drops back. So, but this seems to be something linked, but the extent to which, the proportion of that, I don't know, yeah. But it's longer than just that initial shock of being dehorned. It extends. They move, they don't come back. So over the 2 years.
Thank you, Adrian. Perhaps there's also some considerations you can do when you just look at so many collared rhinos that were never dehorned, and look at what those responses are. And I can't recall anything that are substantial shifts like what you've recorded there, for example. All right, thank you. We're going to move on to Maggie, and she's going to chat to us a little bit about rare antelope. So we're getting smaller. We started with Tim, and we're going slightly smaller, you know.
All right. Well, good morning, everyone. I've been warned we might have some load shedding during this presentation, so hopefully I don't get so lucky. Today I'm going to be talking about some declines in rare antelope populations in the Kruger Park. It's a little bit of a shifted title from what you have on your programs, because I think it can be partially explained by climate drivers. All right, so for those of you who aren't familiar with the park or the antelope within it, in the northern, mostly the northern part of the park, we have 4 locally rare species of antelope, the sable, Tesabe, roan, and eland populations. And in the late 80s and early 90s, these populations crashed. And so this is a problem for park managers, because you want to have a park that has a heterogeneous ungulate population. So one quick sidebar is there's been a lot of work on the effects of artificial water holes on these rare antelope populations, and how these water holes may have brought in more predators that are following the more common species that are moving into areas of the park that previously were inaccessible to them because they're further from water sources. And this is a great figure that I love, showing the incidence of waterhole placement in the park that increases. And then after this crash in the late 80s, park management adapted and started closing some of these waterholes as well. And there's a great paper by Norman Owen-Smith and Lisa LaRue, who are in here, that the provision of numerous artificial water points are really affecting this roan crash in this example, and that maybe these populations are recovering. But you'll note that this paper is from 1998, and if we look at this map again, you can see that these populations haven't really recovered as much as we would hope. So the focus of my talk today is thinking about maybe there's some climate drivers going on. So thanks to Reese earlier today, you heard a lot about climate change and how it's affecting ungulate populations. So probably follow from that. And then the second aim of this talk is actually inspired by a conversation with Sam. Like 2 years ago, he asked, why would you treat these 4 species as a group? Because they have very different life histories and different browsing and grazing strategies, so maybe we can't treat them as a group. Maybe they just coincidentally responded in a similar way. So for the first question, I'm going to focus on a conglomeration of data sets from 1989 to 2011 of ungulate surveys in the park. So a quick sidebar on these surveys is one of the difficulties with working with park data is that there's several surveys that were going on concurrently of the park and then switched over to different methods later on. So first we have an aerial survey of 16 different species in the park, and then there's kind of a concurrent helicopter survey of just the mega herbivores and some of the rare game. And then later on, there was a shift from this very intensive early system of censusing to a much more streamlined transect-based survey. And so what me and my co-authors have done is to kind of translate these surveys, and here I'm using African buffalo as an example because I think it's a pretty good example of all the issues with the data, is to take a measure of effort, how much survey area and time was used to cover these surveys, and get a better sense of the trends in these data. So now we have these animal population counts. We have to think about how they're arranged spatially on the landscape. So I don't expect you to be able to read all of these, but what you can see here is that there's a lot of different landscape types in the Kruger Park. There's 35 that were outlined by Gertenbach in 1973. And so what I wanted to do is to use these landscape types to get a sense of where are these animals on the landscape that are very, not homogeneous, but like the similar landscape cover type. But then to add something about water access as well. So we used distance to river. Here we use 5 kilometers as kind of an average distance to river to break up these shapes so that especially those long ones along the mountains to the southeast, break them up vertically. So we ended up with 166 regions in the Kruger Park. So those are the regions when I talk about regional statistics, that's what I mean there. Okay, so now onto the model. I'm not going to focus too much on the mechanics. You can talk to me later, but just know that I'm using a generalized joint attribute model. Generalized in the sense that it can accommodate very many different data types. In this case, they're all abundance count data, but it could be presence, absence. And then joint attribute means we model all species abundances simultaneously. So in future work, this will be really important when we talk about species interactions, but in this case, it's a static model, so there's no interactions being declared here. And then, on the right there, I have, if you want to read more about the model, but I'm not going to say it. All right, so I have here 6 different covariates that I'm looking at: minimum park temperature, rainfall, land cover, whether the river is within 5 kilometers, whether there was a recent fire in that region, and the number of boreholes per hectare to kind of get at that question of the boreholes. To note here is that park temperature is a park-wide scale, and that all the other covariates are measured at the regional scale, and that some of them are changing by year, like climate and management changes, and then some of them are static, so the land cover and the river distance. Finally, as I mentioned, it's a static model, and also I ran it jointly for 16 species, but for the sake of clarity in this talk, I'm only going to be talking about the rare antelope. All right, so to get back to the first question, are rare antelope population patterns largely driven by changes in climate? I've broken up these covariates into 2, one being climate-driven and one being not so climate-driven, although you could argue that fire can really go in either of these categories, but for now I'm putting that in the management category, and we can talk later about that if you want. So to start with the second one, for non-climate-driven parameters, what do I mean by climate-driven responses? Well, so in this case, the dark red is fire that has occurred in the last year, and the light red is no fire. And so a positive response might look something like this, where you have the species is having a larger abundance in those areas with more fires. A neutral response might look something like this, where you have no correlation between fire and species abundance. And then a negative correlation might look something like this. We can also take this, here's the promised load shedding. It seems my microphone's still working. Okay. So we can also look at this in the sense of a graph like this. So on the y-axis, we'll have the species response. It can be positive, neutral, or negative. And I was going to show you these graphs for each of my covariates, and then I thought that's a little bit confusing and too many graphs to show you, so I'm only going to do it for a couple. The first one being fire, because I think it's important to note that while we have all 4 of these rare antelope species that we're looking at have positive responses to fire, everyone else does too, which is something that we expect, right? You know, you have a fire and it increases the nutritive quality of the grasses or the forbs that are coming in. So ungulates like fire, we know this. But then I thought, maybe I'll show it to you like this. So you have the 4 species that we're looking at, and then the column is going to be the covariate, and whether it's positive or negative response, and they'll be a little bit grayed out if it's not as significant as, so for example, all of these are highly significant. Okay, so, and then responses to rivers. Sable and Sabi, not much response, but Rhone seem to avoid rivers, and Eland seem to be attracted to rivers. You can remember that for later when we talk about land cover as well. Boreholes, this was a little bit more interesting because given what I had said before, it seemed like boreholes are really driving these species crashes. But one thing to note is that the timeline that I'm looking at is mostly post-crash. So I'm thinking, you know, perhaps it drove the crash in the beginning, but the suppression of antelope populations afterwards maybe wasn't as driven by borehole densities, or maybe removing some of those boreholes in the park was helpful. Lastly, for those non-climate drivers, I have land cover, and so I took those original 35, kind of broke them into broad categories so that they're a little bit easier to understand and model. You don't want 35 categorical variables in there. So first to note is that the alluvial area, which is mostly near rivers, again, we see eland positively responding to rivers. This is to be expected. For shrubby and sparse felt and Mopani woodlands, we see that all of the rare antelope responding positively, except for sable to the shrubby sparse felt. And then for all the other land cover types, there's a negative response. Although I will also point out, again, that fire, all the other ungulates had positive impacts. And then also for Sandyfeld in the Pundamiria region, there's just low density of ungulates in general. So those two aren't really indicative of the rare antelope. Okay, and then on to climate drivers, which is more of the interesting thing here. So first we have temperature, and so I put a graph up here so you can see the temperature increases. One thing that anyone looking at this would say is, well, isn't temperature then just a proxy for time? And I say, yep, you're probably right, so that's moving forward, something I'm gonna think about. But in general, all the rare antelope had negative responses, and again, going back to our box plots, you can see that actually they're quite shifted out as a group, and that most other species are responding positively to increases in temperature, which I thought was quite interesting. And then with rainfall, we see negative responses for all but the sable, which makes sense because one thing I didn't mention is that most of these species only live in the north of the park, except sable is pretty widespread at low densities everywhere. So, conclusion for this first section. For climate, yeah, we see some evidence that there's some climate effects. And for non-climate, it's a little bit more of a mixed bag. Land cover type is really, really important to these species, as many of you know. So in conclusion for this first question, rainfall, temperature, and land cover type are the most important. But maybe we need dynamic models to tease out climate as a main driver. Some limitations to mention. Again, I said the correlation between increasing temperature over time. Also, temperature here is a park-wide statistic, so I want to get into more region-specific measures. Static models don't account for year-to-year differences, so in the future I'm going to be running a dynamic time series model. And then also to mention, this is the paper with Norman and Lisa. It was about the Lee effects in sable populations. So this is when some populations are at very low densities, then perhaps they have trouble recovering over time, even though conditions improve. So you can check that out if you want. So answer to question one, likely yes, but we need to model dynamically. And so that's going to be the bulk of my second chapter of my dissertation, so stay tuned. All right, and then for the second question that was inspired by Sam over here, is this a coincidental decline? Let's find out. So we're gonna, the way that you read these results is that this is not gonna be a phylogenetic tree, this is using a correlation distance. So species A and species B here are more closely related than species X and Y are because they have a shorter distance on this X-axis. And they're more closely related to each other, kind of grouped together, than they are to between groups. And so if we look at just the raw data, like where are these species occurring on the landscape, you don't really see, you see some clustering of the rare antelope together, but it's not as clear of a picture. So this is just, how are they using the space? But if we look at clustering species by their responses to covariates instead of their spatial use, then we see that these species do cluster together. And I thought that was quite interesting to see is even if we change the covariates, I think I dropped temperature at some point because I thought, there's the strong temporal variance there. We still see this clustering of rare antelope. So in response to Sam's question, I think they do respond in similar ways to the environment, and maybe this classification of rare antelope is something more than just species that all declined at the same time. So in conclusion, some takeaways. There's evidence that climate change may be playing a role, but we need to do some dynamic time series modeling, so stay tuned. And that similarities in population patterns are due to more than just co-occurrence on the landscape, and they truly are in some way responding to climate and non-climate drivers as a group. And with that, I will thank all of my co-authors and sponsors, and you can find me email, Twitter. Yeah.
Thank you, Maggie, and great. We have time for one question. Oh, here we go. Tony.
Thanks. So I think something that's really important when you're trying to work out the importance of these, potentially correlated predictor variables is a mechanism. So my question is, what on earth would the mechanism be for a fairly minor increase in minimum average temperature to affect, the population of these species?
Sorry, can you repeat that question? I'm noticing that speakers have a hard time understanding from the microphone.
Sorry, yeah. So my question is, what's the mechanism why would a fairly small increase in minimum temperature affect these species?
Sorry, I really didn't hear that. Maybe you could repeat, Sam.
He's asking why would a small increase in temperatures changing this species? Did I get that wrong? Right.
No, okay, yeah, that's a good question. And I don't know as much about these species' life histories as I would like, but I would think that it's probably some of the results I've shown here are strongly temporally linked. So I think that the strong correlation that you see there that I showed is most likely due to just increasing temperature over time. So I don't have an answer to your question, but probably it's not going to shake out to be as important as I made it out. So thank you for calling that out.
Thank you very much, Maggie. I'm sure you and Tony can have a chat over tea. All right, we're now going to the things that big animals need. You know, they need some food, and they will have some consequences on those. So Duncan is going to talk to us a little bit about reintroducing extinct megafauna and what sort of effects it may have on plant communities. Thanks, Duncan.
Huh. How many of you have watched Jurassic Park? How many of you have watched it 20 times? So I'm not the only one who is weird. I want to talk a bit about reintroducing extinct megafauna and what the implications that could have. And when we talk about rewilding, we're really talking about a fancy term that describes conservation efforts that are basically aimed at restoring and protecting natural processes and wilderness areas. And I know that doesn't help because we don't know what natural spaces are. We also don't know what wilderness areas are. So it's really complicated. It's a field that really borders very closely, or rather brings two disparate fields together. The field of ecological restoration, when we are rewilding, we are thinking about how we are returning things to what they used to be, so that's a bit of restoration, and also a bit of conservation, because really conservation is about reintroducing species to ecosystems, and really the emphasis here is on ecosystem processes. So really, today I just want to focus on these two questions, really question around what does the world look like, or how would the world look like if most of the herbivores, large herbivores and cattle went extinct. And that's tied to the second question, what lessons or rather what can we learn from that about rewilding endeavors? The first question is kind of easy. We can go to these systems where herbivores have gone extinct for a long time, and we can understand what the vegetation looks like. And perhaps that's as a consequence of having these herbivores out of the system for a long time. But also, we can set up herbivore exclosures to exclude different herbivores, and that's kind of what most ecologists do, that you fence out some areas and observe them over time, with the hope that the changes you see are as a consequence of removing certain groups of herbivores. And we're also doing that in our national parks, where we exclude certain land use practices, for example, cattle, and just retain wild herbivores. But then our exclosures is a question about our exclosures really mimicking these processes in the right way. So I'm going to talk about some insights from one of the long-term exclosure experiments we are doing in Laikipia ecosystem in Kenya. It's one of the long-term exclosures based at Impala Research Center. It's now we're counting 27 years since these exclosures were set up. And the idea of this is you go to an area where you have an open space to begin with, no fences at all, everything comes in. And this is a system that also has cattle. So you have all the large herbivores and wildlife species together with cattle. You set up a fence that excludes only the elephants and leaves everything else. And then another set of fence that excludes all the wild herbivores, retaining only cattle. And for the same system, there is another set of plots that now have all the same combinations, but without cattle. It's really replicated 3 times, each of these treatments. So we have a total of 18 plots, each of the plots being 200 by 200 meters on the side. That's for hectares. The plots themselves are located within this nasty soil ecosystem, the black cotton soils. And these are widespread soil ecosystems. And if you know something about this, they have high clay content, around 60% of clay and less sand. If you own a piece of land in these soil ecosystems, you are lucky to sell it when it's wet, because when it's wet, you are dealing with a bigger piece of land because it expands, and when it's dry, it shrinks, so you are dealing with a small piece of land. They have deep horizon because of, again, of the cracking and mixing up of the soil, and they are dominated by one tree species. Again, another good place to be a botanist, because if you walk in, you can almost identify 90% of the trees that you are going to encounter, because it's only one of these tree species that dominates. Perhaps arguably one of the most well-studied tree species, well known for mutualism, and I think other presenters earlier than me have been talking about this, as pines are good for protection for some herbivores, and also in addition as ant species that also aggressively defend us against herbivores. Well, a good story, but gets a bit complicated because we know that not all these 4 species of ants are equally good at defending herbivores against browsing. To some extent, even the lower one there is actually the least defensive and does not live on the tree it lives on the cavities that I'm showing on the side there, and not on the soil thorn demetia like the other 3 species. So this is ideally how it should work. The tree provides shelter in this soil thorn demetia, but also provides nectar through extrafloral nectaries. And in exchange, the ants protect the tree against herbivores. So what happens when you exclude herbivores in this system? Your guess might be as good as mine. Those that have observed trees, you realize that when you remove herbivores, the kind of induced defenses against herbivores sort of start disappearing. And this is the first thing that we saw after setting up exclosures. 2 years down the line, the spine length decreased for trees that were protected against herbivores. Well, if you are a naive herbivore like William, you are lucky because you can browse without wondering about spines. But elephants don't care. They can chew those spines as if without any trouble. The other thing that we found out later, much later, is that also in plots where you have excluded herbivores, you tend to get less of these swollen thorns where the ants live. So trees kind of relax production of those swollen spines. But again, another unexpected scenario here is that the nectaries that were meant to feed the ants, again, after excluding herbivores for a long time, you get less and less of those, meaning that trees are also, again, relaxing production of nectar to feed the ants. Consequently, the ant mutualism collapses. in big ways. And look at this too. This is the species that I told you is less effective in defending. And what happened? It actually increased. And the most aggressive defense sort of reduced. Again, other things that we are observing much later is the sense that also these plots are becoming more dominated by tree species that are or rather tree species that you don't find elsewhere in the open spaces. So they are naive sort of trees that are increasingly suppressed in the rest of the landscape, but excluding herbivores, they do increase. And this is what they are going to do. Whenever they get in, they are picking on trees, and anybody who is running exclosures knows what that means. Anytime they go, they are picking on those tree species that they would not find elsewhere. And they're also class-specific. They are going for the bigger trees and sort of ignoring the smaller trees. Other things we are observing is just the idea that in plots where you have cattle, or rather, the idea that when you remove cattle, perhaps you'd have expected you get much difference, but it doesn't really matter whether it's cattle or wild herbivores that are grazing. What really matters in this sense, in terms of herbaceous productivity and also species composition, is the grazing density. So, again, another unexpected situation there. So really, as we wind up, what are we learning from all this? One of the things that is clear from this is that individual plants relax their defenses and sort of become naive when you exclude large herbivores. And this is happening at individual species, individual plants level, but also could happen at community level, that even at community levels, you sort of get species that are less prepared to deal with herbivores when you exclude them for a long time. And we could really worry what is happening on long-term scales, evolutionary scales, and you can only expect that after excluding herbivores, or rather after herbivores going extinct over a long time, there are dramatic shifts that even could point to lack of constitutive defenses. So really, I want to conclude by saying that native communities that we observe in most of the systems around could be really different from what they used to be before the herbivores got extinct. And in a way, we suggest that these naive individuals may be less prepared to deal with reintroduction of herbivores, and we need to rethink as we think about rewilding and as we think about Jurassic Park, whether the plant species that we have are prepared to deal with the kind of herbivores that we are introducing. We're planning to reverse some of these treatments, and hopefully when I come and we'll have done that, I can share with you some of the insights about such reversals. Thank you.
Thank you very much, Duncan. I'm sure some of our colleagues that are working in exclosures would want that video so they can practice on the way to get in and out of the exclosures. There is not a lot of time for a question, but I'm sure that there'll be some discussions and some of the ideas that Duncan has raised here. We're going to move on to Marcin, that's going to talk to about brown world forests. And yes, Africa is going to teach Europe something. Thank you.
Hello, thank you very much. So I'm the Polish guy who's studying ecological concepts from Africa in the temperate zone of Europe. And I have never been to Africa. This is my first time, so it's super cool. It's wonderful to be here. So in my talk, I'm going to show you how some concepts, ecological concepts developed in southern hemispheres were helpful for us to understand our temperate zone ecosystems. So I come from Białowieża Forest, which is the most famous old-growth, broadleaf temperate forest in Europe, often set as a reference for other places like that. And the dominant paradigm is that Białowieża is the closed canopy forest driven by the succession. So on the productive soils, we should always end up with the forest. And the major process that is driving the dynamics of that forest is related to gap formation. So whenever a large tree like that or several trees like that would fall due to windbreak, there would be opening in the canopy, allowing light to penetrate, and then this allows regeneration of trees to recruit to the canopy. And that implies that juvenile trees that are waiting for this regeneration, they should have traits mainly associated with the competition for light. However, this model doesn't really fit when you start thinking about large herbivores. We have them there, but this model, the closed canopy model, does not really include them or integrate them well. So their effects are actually quite poorly understood and integrated in the temperate zone forest ecology. Moreover, climatic parameters of Białowieża forest actually position it in the ecosystem's uncertain area with its 630 millimeters of annual precipitation per year. So this is strongly suggesting that we might have other alternative stable states possible, not only forest, which is a fascinating and very inspiring idea for an ecologist working in Białowieża forest. And this also, in a way, is strongly challenging the forest canopy paradigm. So this is also the reason for me to reach out to all these beautiful places around the world where alternative states dependent on presence or absence of plant consumers could be found, and also a great reason to visit Kruger National Park. So to exemplify this is Białowieża Forest, a strict reserve, and we have that open spots present there. But they are usually thought to be unnatural and degraded forests because they are usually coming from the abandonment of agricultural fields or other activities of humans. So these patches do not fit the closed canopy model at all. But these places, they have very unique and diverse plant communities, including spiny species and flowering plants and grasses. So forest succession, on the other hand, is also slowed down way more than we would expect from the productivity. So that suggests that there are maybe other drivers than productivity, soil, and moisture that could actually maintain these places open. This is relatively small scale, but already give you a nice example. So the overall goal of our work in our group is to try to apply the Savanna trait framework to understand better our ecosystems and vegetation types in Europe. So this all also implies that we are lacking solid integrative framework to study herbivores, plant traits, and resources altogether. So we borrowed demographic bottleneck model from Africa and adapted it to our work. Importantly for us, this demographic bottleneck model nicely integrates the effects of consumers and resources on woody plant recruitment. Very important for the gap dynamics when you think about it. So this is just a quick sketch of how the system looks like. We have about 100 woody species, 24 trees, 60 shrubs. We have 5 large ungulates, from omnivore to grazer, browser, European bison. We have also 2 predators, wolf and lynx. So this thinking led us to several intriguing research questions that I'm going to show you later. Mainly, are juvenile trees adapted only to a competition for light? And if so, what is the role of large herbivores, and how do these trees cope with the large herbivores? And then we used this framework of traits that should be displayed in the canopy forest, which we refer to as green syndrome, with elongated stems, low branching density, and highly contrasting trait syndrome coming from the savannas, where you would expect more frequent forks, short internodes, smaller leaves, and so on. So we worked with that framework in our research. So back in 2008, we set up a common garden experiment where we planted 5 dominant tree species in our forest that also differed in their initial juvenile architectures. And then we controlled, so we planted them in combination of 4 treatments, controlling light. So we put, we put plots in the gap in the closed forest, and we also controlled herbivory, so we put exclosure and the unfenced control in these places. So what did we find? First of all, we found strong interactive effects of light and herbivory. And as a result, we found that there are stronger effects of herbivory in the gaps than under the low light canopy. We also found strong interspecific differences in among the 3 species we planted between the plots without the herbivory and in plots with herbivory. So this makes sense because other studies we have done in our forest suggest that gaps in our forest, they are highly attractive for herbivores. So for red deer is utilizing these gaps 2 times more than the plots outside the forest, so they're attracted to be there. So this means that in our system, not only plants, but also herbivores follow the resource gradient. That means that their pressure in these places is much higher than elsewhere. So with this increased consumer control, only the best adapted species could survive, and that leads to species filtering on a relatively small scale, but still. So we went ahead and we tested an idea that branching architecture of a tree could be used as a defense trait in plants and in woody plants, as was shown in African savannas. So we compared changes in branching density among the planted species, and we observed that some of these species develop dense cages in our experiment, but some species were not able to do that in the same experiment. This ability to produce or induce the cage was highly related with their survival probability in our experiment. So to scale up and move away from the experiment, we can observe in Białowieża forest small patches, old gaps, looking like that, where actually browsers can maintain them relatively open. They can slow down succession of trees in these places. They would filter out species that are not adapted. They would allow grasses and herbs to establish, in that way create what we call a brown world, forest patches. Also, this is a place where you could find these amazingly plastic species that actually create cages when they are attacked by herbivore. When they are not, they actually grow as a nomad tree. This plasticity also allows them eventually to escape browsing pressure from a dominant herbivore. Once they build cage big enough, on the middle top there are shoots that are able to escape. If you have seen something like that in your study system, I would be super happy to have an email from you with a picture. So to conclude, herbivores in our system, they strongly regulate or control the number and species composition of the regeneration of trees. In response to resource gradients, the herbivores, they tend to concentrate in gaps. And there they have stronger effects than elsewhere. That could resemble something that you may refer to as grazing, browsing alone, basically. So in principle, only species with traits that are related to herbivore can survive in this plot. I think this is very important for us because it reveals the natural variation of our plants related to herbivory that we have not much understanding of in our part of the world. So I like to think about Białowieża that it's mainly green, okay, but it's also brown, and it gives us nice background and idea to move forward to the real open systems. But obviously, I heard here that fires are really important in savannas. We don't know about them. We don't know anything about them. Fires are completely excluded from my country. Every fire has to be put down and stopped. But we can experiment with fire, so that's what we do. But we do it outside Białowieża forest. We do it in a set of experiments in an associated foundation in Sweden, where we also have a set of exclosures with the common garden experiment, and we also tortured these trees with low-intensity fires, and we already have some cool results. There is more coming, and well, I leave you with that take-home message. I think we need way more African thinking in Białowieża than we have experienced so far. And I would like to thank key people sitting here, Joris Kromsicht, who have introduced me to the Savanna concept many years ago, Tristan, Dries Kuiper, Mats Niklasson, Karin Amsten, with whom I collaborate. Thank you very much.
Thank you very much, Marcin. You've done very well. We've got time for some questions. You've convinced him, eh? You're taking the lessons.
Take it with beer.
Oh, here we go. Here we've got a question. Oh, there's some late awakers.
Thanks for this great talk. Excuse me. I would like to know, do you have some evidence for example, remote sensing and so on, that those gaps are long-lasting or from field?
Yeah, we just have finished collection of old satellite images that became available for free. I don't remember the sensor, but old American satellites that were going above Białowieża since the 60s. And there is a group of GIS experts that are actually looking at it now. So they were interested mainly to look at the effects of forestry. There's lots of logging going on, so that was a major question. But one component that I was also trying to get from them is to actually exactly what you asked, so to get the old gaps. And if we could track these gaps until now, or maybe they're gone, and see the dynamics of how does it work. But this is an ongoing project, and I don't know if we get these results and when.
Sally, please.
Hi, thank you. This is more of a science socio-ecological question. I was wondering how your research community in Poland who works in that forest, how they are receiving your results. Is there kind of a growing group of people who are interested in this in these ideas, or are you a bit of a social outcast?
Well, they usually just stay silent at the moment.
All right, we've got time for one more. I think, Corley, please.
Thanks, Martin. That was a great talk.
Thank you.
I was just wondering, is there not ways to test whether you didn't have more heterogeneity within that larger forest footprint, just using, sediment cores or carbon isotopes, for instance, just to see if your forest was maybe not more patchy going back, in quite a long time period?
Yeah, so there is quite some paleo work done, but they all interpret this data as was closed canopy forest. But I think we need Abraham and his methods and stuff that he's doing here and give him our data to progress. I think that could be a little bit of a game changer in paleobiology here.
Thank you very much, Marcin, and you need to talk to Sally about claiming you're a social outcast of a team. All right, we're going to move to back into Africa, and Tyler is going to reflect a little bit on the impacts of herbivores on some vegetation structure. Thanks, Tyler.
Great. Thank you, Sam. Thanks, everybody, for coming this morning. The question I want to ask today is, does plant diversity matter? And specifically, does it matter when we think about how large herbivores in savannas affect vegetation structure? So vegetation structure and a whole host of associated terms mean many things to many people. I'll take this broad definition for my talk today, which is that vegetation structure is really anything that measures where in three-dimensional space vegetation biomass exists in an ecosystem. And in the context of savannas, there's two things I want to point out. The first is that vegetation structure is a defining feature of the savanna biome. We talk a lot about tree cover as being a really defining feature of this biome that has a lot of implications for the ecology of the system. But tree cover is a structural metric, and it varies widely across savannas, as you can see in these photos here. It's also linked to vital ecosystem functions, not only in savannas, in forests, in other systems, but in savannas in particular, we can think about links between vegetation structure and carbon sequestration, nutrient cycling, and particularly the movement and behavior and diversity of large herbivores. So I like to think this is the elephant that Duncan just showed in his plot. This is what they do. So herbivores have a variety of ways that they affect vegetation structure. At the individual plant scale, animals like this elephant can obviously fundamentally change where in three-dimensional space this tree canopy is. It's getting much lower here. And at the landscape scale, these can, of course, scale up to affect tree cover and overall tree height, forage accessibility, that sort of thing. On the other end of the spectrum, we can think about grazing lawns, a very low structural complexity feature on the landscape, but one that is nonetheless a hotspot for a lot of animals, and that at a landscape scale adds to the overall heterogeneity. So we have some low complexity habitats and some high complexity habitats. And these are maintained by a variety of herbivores, including rhino. If we zoom out into a continental scale, Carla Staver and others have done some really nice work showing that the composition of the large herbivore community can have strong effects on the balance of trees and grasses, so tree cover, productivity, and structural complexity, and that systems with more browsers or grazers can have predictable effects on overall structure. So at scales ranging from individuals to the continental scale, herbivores have strong effects on vegetation structure. But like I said, what I want to talk about today is another perhaps underappreciated avenue by which herbivores might affect vegetation, and that is through effects on plant diversity. So this idea came out of a review that we published earlier this year where we collected a couple dozen studies that used remote sensing, primarily LiDAR, to look at various metrics of vegetation structure, everything from simple canopy height to some of the more complex metrics that are typically used in forest systems, and they measured those across a gradient of species richness. So I pulled out just a few examples here to show what these relationships look like. Across the board, we found almost no evidence for negative relationships between diversity and complexity, so you can see that they tend to be positive, and they tend to fall into sort of two broad categories. There's linear relationships where adding an additional species has the same effect no matter where you are on that species richness gradient. And then one of the more common ones was these saturating relationships, where you have a big jump in complexity at low levels of diversity, but at some point you reach this sort of flatline curve here. And so we talk about this in the review paper as a sort of classic example of a biodiversity ecosystem functioning relationship, where you have different types of functional redundancy in different systems that lead to these different shapes. But we note that one of the biases that could potentially affect these results is that more than 90% of the studies that we were able to find compared this in forests, and typically in temperate forests where you have relatively low overall diversity. And moreover, very few of them experimentally manipulated that diversity. So it's typically comparing forestry sites that have different forestry practices, and so it's difficult to disentangle the effect of species richness from some of those forestry management practices like selective logging. And so what I'm going to talk about today is an attempt to fill some of those gaps by looking at how plant diversity affects structural complexity in a non-forest system. This is the Savanna at Impala Research Center in Kenya. A different exclosure experiment than the one that Duncan talked about. This is the Uhuru experiment you can see in this drone photograph. And I'll just point out that it is it's a dry savanna, and this is a photo taken after 2 years of drought. And so that's why you're not seeing a whole lot in the grass layer there. But I'll just ask you to keep this in mind, and particularly some of these differences, right, where you can see there's a thick grass layer in these plots and not here. Duncan did a great job of introducing why we do these sorts of exclosure experiments. We're simulating herbivore extinction in the landscape. Just as Duncan showed, we have a variety of treatments in each of these plots. So with the 36 total plots spread out over a pretty significant rainfall gradient. There's an unfenced control that allows access to all 22 species of wild herbivores at Impala, and then the other extreme here in the back is a total exclosure where nothing gets in. And then there's 2 intermediate treatments, one that excludes just elephants and giraffes, and one that excludes everything larger than dik-dik and warthog. And so the nice thing about this sort of experimental design is that not only can we look at sort of the comparison of the control to the total, so what would happen if all herbivores went extinct, but you can do pairwise comparisons between, for example, the control and the mega plot that tells you just the effect of elephants and giraffes. We have a very rich data set from this experiment, which was initiated in 2008. We just published a second data paper, so all of this data I'll be talking about today is publicly available. There are 450 species and counting of plants in the herbaceous layer. These are surveyed biannually in about 1,800 1-meter squared quadrats spread out throughout these 36 plots. So that's a very rich data set at a very fine spatial scale. The tree community is more diverse than the one on the black cotton. We have more than 40 species of trees, and these are surveyed at a slightly larger scale, so 10 by 10 meter subplots, and there's about 1,300 of those spread across these plots. And finally, at the end of the talk, I'll talk about how we use LiDAR that we collected, LiDAR data that we collected in January of last year to look at some of the structural metrics in these plots. So the overview of the talk, I guess my hope today is to give you brief answers to 5 related questions, and in so doing, hopefully convince you that there are multiple pathways by which herbivores can affect vegetation structure. So we'll start over here using primarily field data to look at how herbivores affect tree abundance and diversity, and how they affect herbaceous abundance and diversity, and then how some of those changes, particularly in diversity, affect vegetation structure, and then finally at the end, looking at how herbivores directly impact individual plants. So try to get an idea of why we see some of the variation in vegetation structure that we do in this exclosure experiment. Okay, so let me orient you to this slide, because you'll see this on this and the next slide. So I'm showing here, here we're looking at herbivore effects on tree abundance and diversity. Across the x-axis in all these plots is going to be those 4 treatments ranging from the unfenced control, so all herbivores, to no herbivores. And on the y-axis, there's various metrics of abundance and diversity. So again, here we're talking about trees. You have 2 abundance metrics here, tree cover and tree density. And then on the diversity side, I'm just simply reporting species richness on that subplot scale. So I'll point out a few things here. The first is that, as you might expect, elephants and giraffes have a strong effect on tree abundance, on tree cover. So we have a very low tree cover across the board in these plots in the presence of all herbivores, but that nearly doubles after 14 years when you exclude elephants from the system. Interestingly, that tree cover change doesn't seem to be driven by changes in tree density. So across the board, we see similar levels of tree density, suggesting that the primary mode by which elephants are having this effect is by changing individual tree size rather than the number of trees in the plots. And I'll just point out that maybe not what we would expect, but that is that even the smallest herbivores have a strong effect on the diversity of the tree community. So the only difference between these minus meso and minus all plots is the presence of dik-dik, and yet there's sort of a positive trend here across the species richness gradient where excluding even those smallest herbivores has an effect on tree diversity. If we look now down low in the understory, the results here are a little bit more idiosyncratic. We see this sort of sign pattern here in species diversity. Now I'm showing diversity on the right and abundance on the left here. I have thoughts about why this is. I'd love to chat with people about why we might see nonlinear responses, particularly in the herbaceous layer in terms of diversity. But interestingly, elephants, although we think of them as being these animals that eat primarily in the trees, do have a strong effect on herbaceous layer diversity. But across the board, I think the takeaway message here is that as we decrease the herbivore pressure, we get an increase in herbaceous biomass as well as diversity. Okay, so to this point, I've showed you that in this particular system at Impala, when we exclude herbivores, we get more trees, more herbaceous vegetation, and the diversity of those two plant components increases. And now I want to just talk quickly about some structural metrics. This is what you're looking at here is a canopy height model from our LiDAR data at 25 centimeter pixel resolution. It's colored by the height, so the yellow blobs are trees. And the nice thing about the approach that we're taking is that we have sufficiently high resolution that we can actually look at these meter scales to get at structural complexity. And so what we did, and I'll gloss over the details here, is we can essentially take the whole LiDAR data set to look at tree complexity, and then we can just peel away the tree layer and you can still get complexity in the understory. And we have good field data to sort of calibrate where we make that vertical cutoff to separate these two layers. Okay, so there's a variety of ways that you can measure structural complexity. These are all primarily used in forests, and so there is a question about how appropriate they are to adapt for savannas. But here we're looking at the effects of tree diversity. So in each of these figures across the x-axis, we go from 0 tree species richness to 10. This is again at that 100 square meter subplot level. And we have a few that you'll probably be familiar with: maximum height, mean height. Coefficient of variation is from the raw LiDAR data, so how variable in vertical space is tree biomass. Rumple and rugosity are common forestry metrics that essentially measure how rough and kind of wavy the top of the canopy is. And then percent cover, you'll all be familiar with, is just the ratio of kind of filled pixels to unfilled pixels. And these relationships across levels of diversity fall into a few broad categories. We have these four, which display a sort of saturating curve, and especially a big jump between 0 and 1 species, right? So as soon as you have something there, as soon as it's not an empty savanna, you get a lot of that structure. And as you add more and more species, you see very little change in some of these metrics. Something like percent cover has a bit more of a linear increase, where even in this sort of range where you're going from 4 to 5 or 5 to 6 species, you're still seeing a pretty significant increase in cover. And then rumple, this kind of weird forestry metric, is one that actually has a negative relationship. And this is, I think, a great example of why we need to think about how structural metrics can be applied to savanna systems, where in forestry we think about more trees as being more complex, but in savannas that's not necessarily the case. Now, if we zoom down to the understory, the patterns here are broadly similar. We see positive relationships between herbaceous species richness here, ranging from, again, 0 to 10 herbaceous species. And I think the real key takeaway here is that with this high-resolution LiDAR data, we can essentially treat the herbaceous layer, this layer that is below 1 meter, as a mini forest. And so it's sort of like the mouse's eye view on what an herbaceous community would be like. And what we see is that the same dynamics play out. If you add more species, you get more complexity. And you can think about how some of these different metrics might apply to different species and what we want to know about animal movement and microsite selection and that sort of thing. Okay, so finally, I'll present just a little bit of field and remote sensing data to answer this question about how herbivores affect vegetation structure directly. You'll remember that I said that elephants affect percent cover, but they don't affect the density of trees, and so I suggested that might be because they are affecting the size of individual trees. So here we have canopy area. This is field measured data on a long-term data set of individual tagged trees that have been tracked through time. So these data are from 14 years without elephants. And you'll see that for these two species, these are the 5 most common tree species in this habitat. For these two, which are among the more common of these, you can see that there's a big jump in canopy area between the control treatment, where especially Acacia brevispica is very small, to that minus mega treatment where we've excluded elephants. So field evidence that the mechanism by which elephants affect percent cover is through changes in canopy size. So this is a horizontal change in the structure of the vegetation. We can also look vertically using LiDAR data. So I pulled out just one example of that here. We can segment individual trees from the LiDAR data and then look at the distribution of points vertically. And this Acacia mellifera makes up about 40% of the trees in the diet of the herbivores in this particular part of the system. And you can see, especially for the elephant effect here, the effect that elephants are having is decreasing the overall height and also concentrating tree biomass here at about the 2-meter level, where it's going to be much more accessible to other herbivores. So I'll just finish by saying that hopefully I've convinced you that herbivores in this system reduce the diversity across the board for both trees and herbaceous vegetation. More diverse plant communities tend to be more structurally complex, but I think we need to think about what that means in a savanna, because a lot of these complexity metrics would tell you that it was quite complex if it turned into a forest, and that's not something that any of us want. And then finally, when we think about, as Duncan said, these herbivore exposure experiments as an example of extinction, you need to think both about direct effects, those elephants eating trees, as well as indirect effects, and how those are ultimately going to change the structure of savannas into the future. So I'll just conclude by thanking folks who helped in the field, and if this sort of work is of interest to you, particularly for students, I'm opening a lab at Notre Dame next year and would love to chat with folks about opportunities for both graduate and postdoc research. So thank you very much.
Thank you, Tyler. You've used your time very well, so I'm sure there's some people that can chat to you over tea time. All right, we're going to go to Maggie, who's going to talk to us about demographic responses of trees to variation in herbivory. Thanks, Maggie.
All right, hi everyone. So today, I'm a PhD candidate at the University of Florida. Today I'm going to be talking about some of my dissertation work looking at the impacts of different herbivore guilds on savanna tree demography. So we know that herbivores are important drivers of vegetation structure and composition, as we just saw, in savannas. And we also know that across many savannas, mammalian herbivore populations and communities are changing, whether that's a loss of herbivores due to things like habitat loss or overexploitation, or rapid increases in herbivore abundances in protected areas. So it's really important that we're able to fully understand and be able to predict the responses of plant populations to these changes in herbivory. So of course, there's a tremendous body of literature on the impacts of different herbivores on savanna plants, and particularly trees, which I'll focus on today. And we've heard about some of that work today. So we have a pretty good general understanding of the effects of different herbivores on savanna plants. But we also know that there can be substantial variation in these impacts across systems. And this can potentially be attributed to a number of different factors. For example, we might think about how plant life stage or size class might moderate the impact of herbivores on plants. So you might expect that mega herbivores would have large impacts on large adult trees, while smaller herbivores like impala might have stronger impacts on smaller plants or early life stages. We might also expect these impacts to vary depending on the plant species, and this can be for a number of reasons. For example, differences in palatability, structural defenses, resprouting ability, things like this that might moderate the effects of herbivores on trees. There might also be reason to think that herbivore impacts might be different depending on the demographic process that we're looking at. So we often think about the effects of herbivores on tree survival, and we generally think of them as limiting survival, though we can also think of cases where they might facilitate survival, such as with grazers removing grass biomass. But in order to understand the responses of these tree populations to herbivory over time, there's another component of these population growth that we need to understand, and that's recruitment or fecundity. And so we generally might also think of herbivores as limiting this, but there are also a number of ways in which herbivores could potentially facilitate recruitment, such as seed dispersal or aiding in germination with gut passage. And so our goal is to understand the responses of savanna tree populations in terms of both of these demographic parameters to changes in herbivory. So our questions were, which factor best explains savanna tree survival, herbivore guild, tree species, tree size, or some combination of these factors, and which factors best explain recruitment. To do this, we used yet another exclosure experiment. This one is located right here in Kruger, in the southeastern portion of the park, near Lower Sabi in the basaltic supersite. And so at the site, we tagged all individuals of 3 common tree species, Senegallia nigrescens, Vichelia gerardii, and Sclerocaria borea. So we tagged all individuals of these species across 16 plots, and our plots were divided across 3 treatments, and so similar in a lot of ways to the ones that Tyler just described, but we had our open control plots. These allowed access by all herbivores. Then we have these partial, in the middle here we have these partial exclosures, which have our fences that just exclude mega herbivores, so elephants and giraffes specifically. And then we have these full exclosure fences that exclude everything larger than a hare. So we tagged all individuals of these 3 tree species on these plots in 2019, and then we went back and found these plants and researched the plots for any new individuals in 2020 and 2021. And we used these data to estimate survival and recruitment rates. And so what we predicted was that as we sequentially remove different herbivore guilds, so as we go from all herbivores, so small, medium, and mega herbivores, and then when we remove mega herbivores and then remove all large mammals, we expected to see generally an increase in survival. Similarly, we expected that on average we would see an increase in recruitment. However, we also expected to see differences in survival depending on the tree species and also tree size. And we also expected to see differences in recruitment across tree species. And particularly with recruitment, potential changes in the magnitude of these effects and potentially also the direction of effects. So we analyzed these data using a capture, mark, recapture framework. And we did this in order to estimate detection. And so this is just a sampling of studies that have estimated detection for plant population analyses and the potential impacts of imperfect detection on the estimation of demographic parameters. And we thought this might be an issue for us because as you can see here, I'm measuring one of the trees on our plots. And this tree, like many, if not most of the trees that we sampled, were buried in this thick, tall grass layer. So you can see why, despite our best efforts, we thought detection could potentially be an issue. And in fact, we found that detection did vary by tree height and year, so we were able to account for this in our model. So we used a Pradel model, which is a reverse time model that allows us to estimate recruitment, so that's why we chose this model. And we used this to estimate detection, survival, and recruitment. And we allowed survival to, we came up with a set of candidate models for each parameter, allowed survival to vary by exclosure treatment. That's how we tested for this herbivore guild effect, tree species, tree height, and year to account for annual variation. And then we also estimated recruitment, which here is estimated as the number of new individuals per individual in the previous time step. We allowed that to vary by treatment, species, year, and all combinations of those. And we selected the best models for these parameters using AICC. So our results. So based on the most parsimonious model, we found that annual survival was best explained by herbivore guild and tree species. However, it was not well explained by tree size. Year was also included in this model. I won't get into that too much today, but you can see that variation in this graph here. So here we have survival of our 3 different species in each panel. And then on the x-axis here, we have our different exclosure treatments. So we have our open plots with all herbivores, our partial exclosures where we exclude mega herbivores, and then our full exclosures excluding all large mammals. And so here I just want to note the differences in these responses across species. So for both of our acacia species, we generally saw that when we excluded mega herbivores, survival increased. So the commonality between these 2 treatments here is that we're removing mega herbivores. So we could conclude here that mega herbivores are limiting survival for these species, but not so much for marula. Similarly, for recruitment, we found that herbivore guild and tree species best explained recruitment patterns. Again, year was also included in this model. So here, overall, recruitment was quite low, which we did expect, except for marula, but we didn't see variation in that across exclosure treatments. But we did see a significant effect of exclosures for nigrescens only, and we saw that when we removed mega herbivores, recruitment actually went down. So we've seen so far that both herbivore guild and tree species explained differences in survival and recruitment. And we also see some differences in these patterns across demographic parameters. So I'm just pulling this example for nigrescens. So we have survival on the left here and recruitment on the right. And we can see that when we removed mega herbivores, survival went up and recruitment went down. So they seem to be limiting survival, but potentially facilitating recruitment in some way, although I will just point to the magnitude of this effect as well. So we found that based on our models, this complexity in plant-herbivore interactions really does matter in terms of the responses of these tree populations to changes in herbivory. And so we found that these responses were best explained by an interaction between herbivore guild and tree species. For example, mega herbivores especially seem to have unique effects on tree demography, but these effects are species specific. We also saw differences in these responses across these demographic processes, both in the magnitude of the effect and direction of the effect in some cases. So if we want to fully understand and predict responses of these populations to changes in herbivory, we need to understand something about the herbivore community, tree species, and the demographic processes and how these factors interact. So why does this work matter? I think a major benefit of this kind of demographic data is that it lends potential insight into the future of these tree populations under different scenarios. So for example, this is what our site typically looks like. So very few large trees on the landscape, and there's a lot of interest in understanding what kinds of conditions could lead to the return of large trees to this landscape. And so we can use these kind of demographic estimates to extrapolate with caution over time and look at how survival and recruitment can impact these tree populations over time under current conditions and potentially under different management conditions. And I think also importantly, these species-specific responses suggest that in the future we might see changes not just in individual populations, but also at the community level with potential shifts in dominance of different species. And so with that, I would like to thank everyone that made this work possible and all of our funders, and thank you all.
Thank you very much, Maggie. Yes, there's a bit of time for some questions, so here we go.
2 questions, actually. One is, how do you define recruitment?
Recruitment, in this model, it's the number of established individuals per individual in the previous time step. So it's a per capita measure.
So if I remember correctly, survival goes up and recruitment goes down as you go across that plot. Do they compensate in some way, or do you know, I mean, why does it happen?
Yeah, so that's a good question, and ideally what we would like to do is look at lambda as well and see, but we don't have
because of what this landscape looks like, we don't have all life stages, and so we'd really want information on all of that to look at lambda. So we're not sure. It's a good question. That recruitment result was quite unexpected, so I'd like to dig into that more and see what's going on there.
Thank you very much.
Thank you.
And thank you to all the speakers for having a good session for us here. Before you cheer them, I'm first going to make announcements. Judith has given me a long list here. The very first one is the poster session is all done. Those posters can be taken down if you want to take them with you back home. If you don't, you can leave them here. We always put them up somewhere in our offices and people have some more time.
Hi, everybody. Welcome back to the... we'll be continuing now with the plant-animal, plant-herbivore interaction session all the way until lunch. First up, we have Peter Boucher, who will be talking about some of the linkages between animal diversity and vegetation structure in East African savannas. Thanks, Peter.
Great. Hi, everyone. It's really exciting to be here today. I'm a postdoc in the Davies Lab at Harvard University, and I'm here to talk to you about monitoring bird diversity with UAV LiDAR in an East African savanna. And I will introduce you to these three lovely people in just a sec, and this lovely drone, too. But first, I'll give you some context for this project, because I think it's actually pretty unique. So I think we've been talking a lot at this conference about savanna ecosystem services are generally undervalued, right? And so this project actually comes out of this idea of what if we could value other ecosystem services of savannas besides like above ground carbon trees. Things like biodiversity, maybe the support they provide to communities, those kinds of services that are equally as valuable, if not more. So our role in this is we kind of have a partnership with three entities. It's our lab, and then also a nature-based solutions company called Earthacre, and also an ecotourism company called Game Watcher Safaris. And our role in this partnership is kind of to figure out how we can monitor and map biodiversity at larger scales so that we can start to value those services. And then their role is kind of more to figure out how we can actually value that biodiversity, maybe sell it and find other alternative ways to finance conservation. So that's kind of the background to this project. But we're going to focus on this number one right here. So now you know these people. This right here is Wilson Kasene. He's a ecotourism guide in Kenya, in the region that we work in. And he's also part of the local Maasai community. He works for Game Watcher Safaris. And then we also have Tom, our drone pilot, who's keeping this in the air and being very safe. And we have Viraj right here, CEO of Earthacre. And this sort of partnership comes together so we can learn how to try to figure out how to map and monitor biodiversity with UAVs. So when we're thinking about where to start here, there's been a lot of great research in the past on linking bird diversity to structural metrics derived from LiDAR. So basically, the idea is that there's a hypothesis that the more complex the structure of vegetation and topography in an area, the more diverse the community, the more species, the more niches. And this particularly works well for birds. So what we want to do is actually take a LiDAR point cloud, which measures vegetation structure. Right here, this is a point cloud with a bunch of points with X, Y, and Z coordinates. So you're just seeing a side view right now, but it's 3D. And they're colored by height. So the top right here in yellow is the top of the tree, and then darker points are near the ground. So we're going to try to make some links between bird diversity and bird communities and vegetation structure here across the landscape to see if we can upscale it. So here's more about our site. This is the Selenke Conservancy in the Amboseli ecosystem in Kenya. It's just north of Kilimanjaro, which is right there. And when you zoom in on the site, you can see this blue area right here. That's the Conservancy. It's an ecotourism site, has a very small, high-end, just like 17 tents in the park. And it's part of this larger group ranch outside, which is about 200 acres. There's a bunch of Maasai communities who practice grazing there. But this park has been set up for about 30 years and managed, and the access isn't fenced, but it's managed and protected. And so you'll notice some other ecological gradients in here that we might need to consider when we're trying to map biodiversity. One is soil type, which you can see in the Google Earth imagery right here. So you have black soils and red soils. And we also have this gradient that we're actually really interested in, right? This sort of inside the conservancy where it's protected and outside the conservancy where it's generally considered to be more degraded. There's a lot of invasive species, a lot more bare ground, just a lot of overgrazing is essentially what's out there. So we have sort of this gradient of diversity, and we can sort of use that to try to get a mixture of vegetation structure and diversity relationships. So what we did is we did some UAV LiDAR vegetation surveys. We covered all of the area that you see outlined in white here, and I've also overlaid these green points right here. Those are actually, that's tree height So it's showing you where vegetation is clustered. And the white areas are actually a terrain model from our LiDAR data. So that's what's overlaid. So everywhere in white is where we sampled. And then we also had these bird surveys that were collected by Game Watchers guides. It was a huge effort on their part, and really amazing data was collected. We have 10 transects, 50 sites, and they're split evenly between black soils and red soils, and then 60-40 inside and outside the conservancy. They took bird observations, sights, and sound at dawn and dusk, and and then we used some of those metrics that were generated from them, so diversity metrics from that bird data, in order to relate it to vegetation structure. So just giving you a little context as to, do these communities actually vary across these gradients? This is an ordination of the bird community data. So points that are closer together, each point is a site. If they're closer together, it means they're more similar communities. And you can see it splits pretty evenly, or it splits pretty well along the soil types. So there's definitely differences in the communities between soil types. Then if we look at our other gradient, which is sort of inside and outside, we also get a split between the community types. So we have outside the conservancy and inside the conservancy right here. And in both black and red soils, you see pretty big differences between the communities. So that bodes really well for us. Okay, so let's get into the nitty-gritty of how we actually are going to do this, relate vegetation structure to bird diversity. Well, we want to build these metrics, these relationships across soil types, maybe using some kind of vegetation gradient that tracks with diversity. But the one thing that we don't really know is at what scale of analysis does this actually work? So do you have to relate bird diversity collected at a point from trees and grasses that are 10 meters away? Is it 30 meters away, 50, 100? We don't really know until we investigate it. So that's where we came up with this study design, where essentially at every single site, we take varying radius plots from the center of each site, and we grab LiDAR data at each one of those radiuses, and that's one picture is right here, looking at top down, just a circular area. And then we generate vegetation structure metrics for each one of those plots, run them all through models, and try to figure out which ones work best. So just to give you a sense of like what kind of vegetation structure metrics are we measuring? Well, there's quite a lot. There's 114, some of which are grabbed from the literature, some of which I made up because I thought they might work. But a lot of them are based off of this vertical profile of vegetation structure. So right here, if you took this point cloud right here, and say you squeezed it all into the vertical dimension, just looked at point density, which is on this axis, by height, which is on this axis, you can grab some metrics out of that, such as your max canopy heights. You can also look for peaks in that distribution that might tell you where the vegetation is clustered. You might be able to find canopy layers. You can look for other attributes like median height. And you can look at stuff at the differences between canopy layers and the number of canopy layers. This vertical gap size right here, I'll explain later, because it turns out to be pretty important. Cool. So I'm going to show you some results from our relationships between Shannon Diversity Index and bird diversity. Shannon diversity of birds and vegetation structure. So you have a correlation right here with Shannon diversity of the bird communities on the y-axis, and you have your plot radius right here on the x-axis, both for black and red soils. And so if going from left to right here, you're getting bigger and bigger with that circle, that distance from the center of each site. And I've grabbed the top variables for each soil type. You'll notice that they're different, actually. So red soils and black soils seem to have different variables that relate to bird diversity, which is interesting. Although there's some that are repeated, like this gap size metric that I mentioned. Also, you'll notice that they have different relationships. So black soils, the correlation sort of goes up. It peaks at 50, and then at 50 meters, those 50-meter plots, and then it kind of plateaus as it gets bigger. Red soils don't really do that, right? They dip, and then they come up, and it looks like they might even keep going beyond our max distance, which is 130 meters. Also, black soils tend to do a little bit better. It looks like you reach a correlation with variables of about 0.8, whereas red soils, it's about 0.5. So that tells us there's pretty different things going on within each of these soil types, and the spatial arrangement is different. Essentially, black soils probably more homogeneous, so you kind of get up to 50 meters, and then everything equals out and is the same. So we grabbed some of these variables and fit them, fits sort of our final models for each plot radius. So as you go down in this table, this is showing you some of those model fits. I just want to show you as you go down, your plot radius gets bigger, so you're expanding that circle. And it looks like your correlation gets better and your error goes down, which is great. So bigger plots do better. But also we kind of get these two points at 50-meter plots and 130-meter plots where these things tend to work out pretty well. So these two are probably the best places the best sizes to model bird diversity at and make these relationships. And so to tell you about some of these important variables, so vegetation height and layering, represented by this gap size right here, that seems to be really important for both soil types. So actually, this gap size, what it's trying to do is measure the distance between canopy layers in a point cloud. So in order to have a sort of gap size measurement, you need to have multiple layers, and you need to grab the distance between them right here. And you're actually looking at the variation in that across a landscape. It's not just the gap size. So the variation in this seems to relate pretty well with bird diversity. We also have some canopy height indices. There's this one called VDR, or vertical distribution ratio. That actually was grabbed from a paper that modeled bird diversity in Maryland in the US. It's kind of interesting that it works here too. So both of those seem to be pretty significant. And I just wanted to give you an example of what one of these models looks like. So you have Shannon diversity on the y-axis here, and the variation, the gap size, on the x-axis. And so as you go up with gap size, it's actually really interesting. You sort of have this increasing relationship, and then it dips down, right? So your gap size, your variation in gap size gets to be too much, and then you start going down. That said, it looks like it fits the black soil points, which are right here, a lot better than it does the red soil points. But it's hard to know. It looks like they both kind of work here, and at least this metric seems to work across soil types to some degree. So I just want to show you an example of like what we're trying to do with all this, where we want to get to. And we actually want to map the spatial variation and diversity across the landscape using vegetation metrics. So let's say we have an area like this outside of Solenke that's got both black soils and red soils up here. And we fly over, we get some vegetation structure metrics, and then we rasterize those and try to figure out sort of aggregate metrics at a larger pixel scale, so let's say 100 meter pixels, and use the relationships we built to get your Shannon diversity of the bird communities within each one of those pixels. So now there's a whole lot more you could probably do with this, to relate things like inside and outside, to look at the structure of this, how well it tracks, and of course to validate it too, because there's a lot of work we need to do that. Great, well, thanks for listening. I just want to wrap up by saying it looks like we have some relationships that work quite well in savannas, not just forests. And it looks like we have two important scales of analysis here, looking at 50-meter radius and 130-meter radius. And that just speaks to sort of the spatial arrangement of soil types and how important it is to kind of split those up across those gradients. So I'll just wrap up by saying that birds are awesome, but we have a lot of other data on animal diversity, particularly from acoustics and camera traps, that we could use to also build these kinds of relationships and compare if they're the same as bird diversity, different. Yeah, and I'll just point out in this photo, this is Kilimanjaro in the background right there. That's probably the best thing about Solenke Conservancy, so you can see it from right there. Great, well, I just want to say thank you so much to all the Game Watchers guides. They put in tremendous effort into this. We so appreciate it. And also Earthacre for helping us organize this. And all the drone pilots, engineers, everybody who helped us collect the data. So thank you very much. I'll take any questions.
Thank you very much, Peter. Fortunately, we might have to hold those questions off till break. Next up, we have Andrew Davies, who's going to be talking about some large-scale impacts of herbivores in African savanna in terms of the impacts on thermal landscapes. Thanks, Andrew.
Hello. Great. Hi, everybody. We have been talking quite a lot about herbivores, vegetation structure, how herbivores change vegetation structure. And what I want to talk about today is what we're trying to do is thinking about going beyond that. So thinking about how herbivores interact with vegetation and how that then cascades to affect other components of the ecosystem. And we think about that a lot when we think about trophic cascades and the way that predation affects herbivores, which cascades into other things. But when it comes to herbivores and their impacts, there seems to be a lot less attention given to how they also have these cascading effects. And so all of you know in this room, I'm sure, that animals shape global ecosystems. And we've heard a lot about that today at least. And these are disclosure experiments from around the world, which shows really clearly what happens when you simply put a fence around a piece of area. And generally, the vegetation structure increases, the biomass increases. Many of us know this. And we've been using LiDAR data to do this for quite some time. These are 2 LiDAR point clouds, which you've heard a lot about over the last, well, Peter's talk and Tyler's talk and a few others. This is from the Inkushlu exclosure here in Kruger. And you can see really clearly, as most of you know, inside and outside the exclosure, there's really stark differences in vegetation. We have taller vegetation inside the exclosures, more heterogeneous often in either side, and just a whole lot of things that are going on between animals and vegetation structure. And we can use LiDAR really powerfully to look at these in really fine detail at the 3D structure over large, fairly expansive scales. This is from the buffalo camp near Satara, as many of you know about it. And so looking at these, we've been studying this for decades. The next question is, what does this mean for many different things? Peter's talk was about how this affects bird diversity, for example. What we are interested in this is how that affects the thermal landscape. So how are temperatures different inside and outside of these exclosures? This again is from the buffalo camp. And you can see how inside, there's a lot more areas that are far cooler than the outside. And we wanted to quantify this and think about what this actually means and what herbivores are doing to these thermal landscapes. And we've also heard a few talks about the interaction between temperature and animals and all these different things. We all know that temperatures are increasing. But most models of temperature data or projections look very similar to this. These really coarse-scaled, average sort of examples or changes in temperature. And what these fail to detect is what you can see in this picture, where these wildebeest are very clearly, at this time of day in this photograph, selecting for the shade underneath these trees, which you completely miss at these coarse scales, which is what most of remote sensing for temperature changes are looking at. But using LiDAR data, this is the exact same place. We can begin to model microclimates, looking at the interactions between vegetation structure and temperature, understanding that, modeling it, and then we can begin to look at these much more fine-scale differences in temperature, which are actually really relevant for animals, because animals are likely not responding to average conditions, but they're really responding to the availability of places like this, or these ones actually, the scales are the opposite. That's high temperatures, these are low temperatures, and animals are likely using landscapes quite differently. So what we wanted to do was understand these relationships between animals, vegetation, temperature at these really fine scales to understand how animals are shaping these thermal landscapes. So what we did is we went to the exclosure experiments in Kruger, which many of us are familiar with. We went to 6 of them from the north, Rowan Camp, all the way down to the south with Tlangwini and Makashlo. There were 6 of them along this rainfall gradient. 3 of them are on granite, 3 of them are on basalt. So we had a rainfall gradient, we had different geologies that we could compare. And these also represent gradients of productivity, which is important in this context because we predicted that impacts would be higher in low productivity sites because vegetation regrowth is much lower and slower. And so where you have low productivity, we expected higher effects from herbivores. So what we did is this drone, which you've been introduced to a few times, we took this and we flew it across these 6 exclosure sites in Kruger, inside and outside. And collected some thermal data. So what we have on this drone, we've spoken about the LiDAR data a lot, but we also have an RGB camera, and we have a thermal camera that's all integrated into the same system. So when we fly, we collect LiDAR, RGB, and thermal data all at the same time. And so across these exclosures, this again is in Kushler as an example, we have canopy height. These are LiDAR metrics, which we've spoken about a lot. This is plant area index, inside and outside, which is really very similar to leaf area index, how much vegetation there is in these landscapes. And then we combine that with thermal data inside and outside to measure how temperature relates to these variables. And this we did at a 0.5 meter resolution because that sort of matches the thermal camera the best. What we also did is we divided these exclosures into uplands and lowlands. And so we divided over here, this is in Kushla again, we had uplands over here, and then we also had lowland comparisons, again to look at these more localized differences in productivity. So you're looking at broad-scale productivity differences across the rainfall gradient and the geologies, and then local-scale variation in productivity along the hill slopes. I'm not going to get into too much technical detail about the thermal imagery and the remote sensing, but what I do want to stress is thermal data is tricky to work with, and temperatures change very quickly, as many of you know. And so instead of using an entire orthomosaic of the exclosures like we do with this canopy height or things like this, we actually narrowed this analysis down to individual images that we could take. And so we took thermal images which were about 200 meters wide, and we overlaid the ones that we found that were overlaying both the inside and outside within a single image. And these are the candidate set of all the images that we collected. We then narrowed it down to select some of those that have a good balance between outside and inside, as well as a less or not so much overlap, so they're kind of spatially independent of each other. And then what we did is did analysis on each individual image. So these are basically, and we're going to get a lot more into these, but this lighter orange is outside and the pinkish color is inside. And we began to look at relationships between temperatures inside and outside on an individual image basis, because this is an image that is taken at a single snapshot in time. It's exactly the same time. There's no variation in time for temperature to change temporally. There's also no more issues with calibration of the camera and things like this. So we know that these are very directly comparable to each other. Also, what we're concerned about is weather. Obviously, we couldn't fly all of these exclosures at exactly the same time. So we're conscious that there could be variable differences in weather across the times that we flew. All of this was in the summer, in January 2020. And you can see these are the different sites on the different colors. And basically, what I'm trying to show you here is that they had very similar variation in temperature, a bit more variation in relative humidity. But generally, we feel confident enough that these differences are consistent enough. And also, what we're most interested in is the temperature inside and outside, the relative difference, more so than the absolute differences. So then what we did is took all of those data and those images and those days and aggregated them to make these kernel density plots, which are showing you, these are the 6 exclosure sites across the park from north to south. And this is showing you the temperature differences inside and outside of the exclosures. And consistently, what we found is that temperatures are higher outside exclosures. So basically, where we have herbivores, landscapes are hotter. And this was true across the rainfall gradient, across both geologies. And we did find stronger effects of this in lower productivity sites. And what this plot is showing you, these are the sites over here, and this is what we call Cohen's d. It's basically a measure of the effect size of inside minus outside. So the bigger the negative difference, that means there's a bigger difference from the outside relative to the inside. And the larger that Cohen's d, the bigger that effect is. And what you notice is these are the granites and these are the basalts. It's variation between them, and we can get into that. But basically, the lowest productivity sites on each geology, the Letaba and the Roarenkamp exclosures, were the ones with the largest effects at this broad scale. When we break it down across the hill slopes, we actually find the opposite. So local productivity pattern gradients in hill slopes, generally we have the lowlands are more productive than the uplands due to nutrient flows and all these wonderful things. And we found the exact opposite. Generally, we found increased differences in lowland areas relative to upland areas. And it's hard to see here, but these are all the Cohen's d numbers over here. And they're all higher, at least in the granites, in the lowlands than the uplands, meaning there's bigger differences in the lowlands. When it comes to the basalts, it was actually kind of the opposite. But the basalts, as most of you know, are far less variable when it comes to topography. And so the less variation in topography likely leads to lower effects. So then we know, okay, fine, temperatures are different. Herbivores are changing temperature. Now what we wanted to know is how does that happen? And so we then related temperature on the x-axis with different metrics of vegetation. So this is vegetation height and temperature across these exclosures. This is an example of what the data look like inside and outside of an exclosure. And you can see that when vegetation is short, surface temperatures are always high. But when vegetation is tall, it allows for much more variation. We then looked at, well, what is vegetation height inside and outside these exclosures? And no surprise, we have much taller vegetation inside than outside, a more heterogeneous layer. And what that looks like is basically this. This is in Kushli again. This is canopy height model, much higher vegetation, taller vegetation inside than outside. And so there seems to be a relationship between vegetation height and temperature. We then also looked at PAI, plant area index, so how much vegetation do we have? And we see very similar patterns. Where we have low PAI, we have high surface temperatures. And again, increased PAI, more plant biomass, more stuff inside the exclosures than outside. And that's what it looks like again for Nkushlu. You can see much higher PAI. Inside, you can also see a bigger difference in the lowlands, which is likely driving these differences that we spoke about. To quantify this, we then related effect sizes as Cohen's d of basically temperature Cohen's d. So again, the more negative these values are, the bigger that's outside minus inside. And then we had both vegetation height and PAI over here. And this relationship that you can see, well, I can see, but I don't know what happened. But anyway, you can see this very clear negative relationship between the two is showing that when we have an increased difference or large effect sizes in one of these vegetation metrics, that relates to a bigger difference in temperature. And so we can confidently relate these to each other. And when you start looking at these all together, you can see how they relate. So this is temperature in exactly the same image. This is canopy height. This is plant area index. And you can see how these variations sort of correspond to each other in space and, well, in space, yeah. I was going to say time, but that's obviously not true. And so you look over here where there's a big tree, high PAI, lower surface temperatures, for example. The last thing I'm going to tell you about is what we then did is to think, well, how does this differ between woody and herbaceous vegetation? And so with the LiDAR data, we can, as Tyler showed you, basically peel back the trees and look at just the herbaceous layer because we have enough points to do that. And this is PAI, which we're just going to focus on over here. The relationship between PAI of woody vegetation and grass, very similar. The relationship of woody vegetation PAI and grass PAI, inside and outside. You can see there's much higher PAI of woody vegetation inside. Grasses are more similar to each other, but they're more compact and lower in outside the exclosures. But when we look at temperature differences for woody vegetation inside and outside, what's really interesting, to me at least, is that the woody vegetation doesn't vary much by temperature inside and outside, but the grasses outside the exclosures are at much higher temperatures than the grasses inside the exclosures. So herbivores are doing something to the grass layer, reducing PAI, changing the bulk density, the volume, to actually increase the surface temperature. And that really matters when we think about replacing wildlife with livestock across the continent, which is increasingly happening, and we're replacing mixed assemblages of browsers and grazers with grazing-dominated assemblages of cattle, and what that's going to do to the herbaceous layer as well as to surface temperatures. And so when we wrap this up, what we're thinking about is how do animals affect these thermal landscapes? Well, firstly, first point is there's a lot of emphasis going on about how animals are responding to microclimates. And there's a lot of talk about using microclimates in conservation. But this work really shows us that animals are not passive bystanders. They themselves are changing landscapes. They themselves are changing the thermal properties of landscapes, and we need to understand that. And this, as I said, has implications for changing wildlife populations, for the replacement of wildlife with livestock, and what these areas are going to look like under increasingly warmer temperatures and heat wave events. And so with that, I'd like to acknowledge South African National Parks for their fantastic support, especially Isak, he's somewhere here, I'm sure, and Yaku Mo, who was with the Air Wing at the time, that enabled us to fly these UAVs, which is not trivial, I can assure you. But Isak and Yaku were really helpful. As well as many field assistants, Integrated Aerial Systems runs our ROCs and all our legal side of things. And I'm going to shamelessly advertise while I'm here, because I can, that we are hiring a postdoc to not work in thermal landscapes, but to work in Growing Grows National Park with myself and Rob Pringle at Princeton, looking at landscape ecology and carbon mapping using UAV LiDAR like this. So if you're interested in applying, these are our Twitter handles, myself and Rob, where the ad is posted. Or you can obviously talk to me, but please share the word for anyone who's interested in landscape ecology, LiDAR, and carbon. Great. Thank you.
Thanks, Andrew. I think each year the renders of that LiDAR data and the questions that you can ask with it becomes more and more impressive. I think we are going to need to move on to the next speaker, so if there's any questions for Andrew, you can catch him at lunch. Next up we have Patrick Finnerty, who's going to be talking about the use of virtual neighbors to manipulate herbivore foraging behaviors.
All right. I'm going to talk to you all today about plants that smell, herbivores that hate them, and how we can use it in wildlife management and conservation. Now, this work was done in dry sclerophyll forest in Sydney, Australia, but don't panic, I'll get to savannas at the end. So, oh, where's my clicker? Here's my clicker. And this is part of my PhD. So the big question right at the top is, why do we need to manipulate mammalian herbivore foraging behavior? Now, I'm probably preaching to the converted here, but by eating palatable plants, we know herbivores can devastate areas of post-fire recovery and revegetation, cause millions of dollars worth of damage in forestry and agriculture, drive rare plants to extinction, and probably most importantly, push people to the verge of insanity by raiding their crops and their veggie gardens. So, with mammalian herbivores becoming largely overabundant in many protected areas, both within Australia and also globally, herbivory is becoming one of the leading limiting factors in the recruitment and survival of valuable plant assets in these ecological or economically sensitive areas. So current management solutions to these problems hold a number of social and/or economic limitations. Take lethal control, for example. It's not always appropriate, especially for our native species, whereas physical barriers, such as fences, can be really costly, breached, or not suitable for large or inaccessible locations. So we need to start considering some management alternatives here. To do so, I'm going to take a step back. So a palatable plant on its own is highly vulnerable to being found and browsed quickly by a hungry herbivore. But what we now know is if we surround this palatable plant by a neighborhood of low-quality plants, high in toxicity, low in nutrients, covered in thorns, this neighborhood of plants are creating a form of associational plant refuge, protecting this plant, lowering its vulnerability to being eaten by a herbivore. We know this form of associational plant refuge using real plants can work. This is a photo from the work of a colleague of mine showing the protection of a eucalypt seedling in the middle there from the browsing of that swamp wallaby on the side by surrounding it with a neighborhood of paper daisies. Although successful, this kind of approach can be problematic because these neighborhood of plants are always going to compete for water and resources with the plants we're trying to protect. And in ecologically sensitive areas, we don't really want to go around and changing the plant community by planting these neighborhoods. But what we now know is many mammalian herbivores rely heavily on their sense of smell in deciding where to go and what to eat. Work of mine from 2017 showed that swamp wallabies in Sydney could readily locate 1 or 2 buried eucalypt leaves in vials just by using their sense of smell. And work I helped with not long ago showed that African elephants could choose minute amounts of their preferred food choices in these complex matrices of non-preferred food just by smelling. So odor is important for these animals. With this understanding, I'm going to ask the question of whether we can artificially replicate the smell of a low-quality plant to develop and deploy what I'm calling virtual neighbours, a neighbourhood of virtual neighbours around our focal seedling to degrade perceived patch quality to a herbivore and provide similar levels of refuge as what we could using the real plants themselves. Doing this, I kind of feel like the Donald Trump of the wallaby world because all I'm doing is spreading fake news everywhere. I did this work with swamp wallabies in Ku-ring-gai Chase National Park. For all the cricket tragics out there, Ku-ring-gai Chase is 35 kilometres north of the SCG. I did the work with swamp wallabies. Swamp wallabies are a browser, they're a generalist, a mixed feeder. Swamp wallabies are largely abundant in the study area. They're active both day and night. They're olfactory oriented. They're relevant to wildlife management because of the issues I mentioned previously. And they're a really good model problem herbivore, actually. They share a lot of common characteristics with deer or even African elephants in forested ecosystems or savannas. For a suitable focal seedling to try and protect, I use Eucalyptus punctata. Now, punctata is a foundational canopy species in the area. It has a really wide distribution. Punctata is a major habitat tree and a food source for several threatened arboreal species, and wallabies love it. So it's a really good plant to see if we can try and protect. I'm in South Africa, so I needed to take the opportunity to go wallabies. Then I needed to find a suitable low-quality neighbour, one that smelt to us but had no signs of browsing. To do so, I walked transects in Ku-ring-gai, looking for a plant that smelt and had no signs of browsing. And what I did was found Bronia pinnata. Now, although it may look nice, don't be fooled, Bronia pinnata ticked all these boxes. I then ran several preference trials, looking and making sure that my wallabies preferred my eucalypt seedlings over Bronia, and they did 100% of the time, which is great. So before I go forward and start testing these ideas of the suitability or the success of virtual neighbours, I wanted to make sure that the real plants of Bronia could provide refuge to my physical plants, and to kind of gauge the ideas of what foraging scale these wallabies are making decisions at. To do so, I ran a trial with 4 treatments, the first being a neighbourhood treatment, a single eucalypt surrounded by a neighbourhood or 5 Bronia, a single eucalypt next to a single Bronia to test this idea of how many neighbours it might take to make a difference here, a Eucalyptus punctata against the world on its own, and a procedural control of pots and soil. Before main trial, I ran a 14-day pre-trial period to habituate wallabies to experimental setup, and also to get a background index of wallaby activity in the area. And I measured time to first visit in my main trial and time to first browse. Now, for the purposes of today's talk, I'm just going to focus on the latter. What I found was a significant treatment effect. So a eucalypt surrounded by 5 Bronia, a neighbourhood of Bronia, were less likely to be browsed than a eucalypt next to a single Bronia, than a eucalypt on its own, or a eucalypt surrounded by neighbours. A procedural control, pots and soil. Put into numbers, a Eucalyptus punctata alone was 7.7 times more likely to be browsed than if it was surrounded by a neighbourhood, and even a single neighbour was making a difference here. So this tells us that wallabies are selecting at a really fine scale. Even one plant was making a difference here in this wallaby's decision making, so they're picky buggers. So now I can say a real neighbourhood makes a difference, a single neighbour makes a difference, so this puts me in a really good position to start testing these ideas of virtual neighbours. To develop virtual neighbours, I needed to get the odour profile of my Bronia species. To do so, I did headspace odour sampling in the field, where I sucked odour through these little metal tubes, ran it through GC mass spec, which then pushed out this horrific odour profile of over 480 compounds unique to this species. So with over 480 compounds, there's obviously going to be a heap of overlap with species producing similar volatiles in the area. So I needed to figure out which of these 480 compounds were actually unique to Bronia and potentially informative to my wallabies. To do so, I employed 2 rules of reliability. The first rule is that odours needed to be consistent, that is, emitted across the majority of the plants I sampled. And the second rule was odours needed to be precise, that is, the ratio between these consistent compounds needed to be tight between the majority of the plants I sampled from. Using these 2 rules of reliability, I narrowed down my 480 compounds down to 7 informative compounds, which we think are putatively informative for Bronia. I then mixed these compounds together, checked their ratios in this horrific setup here, and I'm a field ecologist, so this took so long, so I had to admire my progress. Before I move on, I've been saying virtual neighbour a lot, so I'm just going to give you a quick anatomy lesson into what a virtual neighbour is. So a 10 ml amber glass vial, where I put my compound mixture of those compounds I mentioned before in, a metal cap with a scepter, which I pierce with a hypodermic needle. On top of the needle, I have a solid phase extraction tube, which is just the most ridiculous name for a clear plastic tube, and a polyethylene frit, which limits the amount of odour that goes through the tube at the top. Virtual neighbour. I then wanted to make sure how long these neighbours would last in the field, so I put them on a heat plate set to 25 degrees in the lab and showed a proportional change in emission rate that was pretty steady over 2 months, which is great. And the final question I asked before I went and put them out into the field was how many plants does the volatile organic compound emission rate of a single vial actually equate to? So I compared the emission rate of a real plant to the emission rate of my vials, and with some pretty complicated maths, we came up to the conclusion that 3 ml of my compound mixture in my vials was equivalent to the emission rate of 1 plant, which is perfect. So then I ran my trials. I looked at my neighbourhood treatment again with the real plants, my virtual neighbours this time, with the eucalyptus in the middle again, of course, eucalyptus alone again, and a procedural control, this time empty vials in pots. And this is what I found. So I found a significant treatment effect. But in this case, a eucalyptus surrounded by virtual neighbours was less likely to be browsed than a eucalyptus surrounded by its real neighbours than a eucalyptus on its own or next to controls. Now, put into numbers, Eucalyptus punctata alone, and I found this absolutely remarkable, was 20.8 times more likely to be browsed than if it was surrounded by these little virtual neighbours, a neighbourhood of them. And it was double as strong as if we used the real plants themselves. I've put live results here as well because this is currently still out in Kringai. I want to see how long I can actually delay these browsing pressures for. So what we think is going on here is that my virtual neighbours are cutting through that noise of 480 compounds, 408 compounds, and are directly informative to my wallabies as to what these plants are and what to avoid. So they're using this information to avoid these patches stronger than they are using the actual plants themselves, which I think is pretty cool. So what does it all mean? Why does it matter? I've shown it's successful in deterring wallabies in Australia, but I'm not arguing this is a silver bullet at all, rather a really exciting new step in the potential development of a new management tool. It's working with animal motivations to find food and foraging efficiently, not against them, compared to fences or noise or light deterrents, which are there to deter animals that are already motivated to move to an area. It avoids the complications of using real plants, as I mentioned before, potentially a cheaper alternative. And I think this is most important, it can be tailored to the species and the environment. If we know the species that problematic herbivores are avoiding, we could potentially replicate this and see if it works in that system. And this is where I get to savannas. What about the best nose in the business? That's what I'm here to do. I want to see if I can replicate this with African elephants here, with Adrian Schrader. So stay tuned. Thank you very much.
Thank you, Patrick. We do have a few minutes for questions, so if there's any questions.
So, okay, in this site, if it's like saturated with wallabies, and then you put out these deterrents, then essentially they're just going to have less food. And what would happen? Would they starve to death? I mean, isn't it kinder just to shoot them rather than to effectively take away their food?
Deter them from food, does that mean, if I'm not quite understanding, so if you saturate the area with odour, the wallabies won't eat, is that what you're saying? No, so I don't think we would put it out to that extent. We're only using it to deter them from certain areas, not an entire area. But no, I don't think Australians would look kindly at shooting to wallabies. Although with my joke, you probably want to shoot them now, so.
Yeah, that was a really awesome talk. I really enjoyed it. I was just wondering, where in the 2 million hectares are you going to put your virtual neighbors?
Where?
If you're trying, where in this 2 million hectares of Kruger are you going to put your vials to try and deter elephants?
So not going to do it in Kruger. I'm going to do it with semi-captive elephants up in Bella Bella in a more controlled approach to see if it works on a small scale first before we go big. Yeah. Cool. Thank you very much.
Thank you very much. Next up, we have Celeste Murray. Celeste Murray, she's going to be talking to us about potential switches in grass communities in response to grazing by mega herbivores. Thanks very much.
Okay, hi everyone. My name is Celeste, and I'm a PhD student with Elizabeth Leroux. And today I just want to tell you about my PhD project and show you some results from, some of the first results from my PhD. So, as we're walking in the field, we see a lot of these magnificent trees with their green leaves, and we see all of these grasses with their amazing inflorescences and forbs with their flowers. So naturally, we want to explore that. So that means we're always testing what this means for the system and for us as a society. However, no one really knows what happens below ground. And this is a problem because then this means we have this bias towards exploring the above ground biomass, above ground biomass, but no one knows what's going on below ground. And this is a problem because what happens above ground does not necessarily represent what's going on below ground. so leading to this biomass scaling discrepancy. And this is especially important in systems exposed to recurrent disturbance because plants have adapted to persist by investing more of their resources into their roots and root structures. So savannas falls just over the 1-to-1 ratio, meaning there is more biomass below ground relative to above ground. However, I think we should keep in mind that this is a global average, and we should expect some variation depending on whether the savannas have more or less trees and whether the grasses are C3 or C4 grasses. And understanding this below-ground dominance is important because it will have consequences for important ecosystem services such as carbon sequestration. And this is because roots are particularly important for soil organic carbon. Carbon is put into the soil to decompose litter and metabolites, with roots being much more via root litter and rhizo deposition. Roots are also 5 times more likely to be stabilized as soil organic carbon relative to the above-ground inputs. So this suggests that in these environments with a lot of below-ground biomass and the relative importance of roots to soil carbon, these areas have the potential to store large amounts of carbon. But of course, we know that the amount of soil carbon is influenced by a range of biotic and abiotic factors, of which grazing is important. And grazing impacts soil organic carbon by either shifting species communities or by directly influencing the roots. So there's been a lot of studies trying to figure out what grazing means for the below-ground carbon. And the results have been quite mixed. Some saying that it's increasing with increased grazing, soil carbon decreasing with increased grazing, and some just showing no response. This seems to be dependent on the prevailing climatic condition and whether the grasses are C4 or C3 or mixed. However, these studies have mostly focused on temperate systems and especially looking at livestock. And they've also focused on sort of exclosure experiments, looking at the extremes, either an area with a lot of grazing or an area with no grazing. So no one really knows what happens in a natural environment dominated by native wildlife species. So the main question of my PhD then is, how does grazing by these native mega mammals influence below-ground biomass buildup in herbaceous species? And then indirectly, what does this mean for potential carbon storage in a fire-prone system? So one would expect then that grazing pressure will lead to the shift in species dominance towards grazing-adapted lawn species, and then this would have consequences for the below-ground plant biomass. So to test this, we took advantage of a sort of natural grazing gradient in Shishlewe-Mfolozi Park. So Shishlewe-Mfolozi Park is in the KwaZulu-Natal area of province of South Africa, and is one of Africa's oldest protected areas, being proclaimed in 1895. This means that this area has been open to these large grazers for about 127 years. Recently, however, they have expanded a section of the park in the southwest in 2019, meaning this new area has been opened up to large grazers for about 3 years. Then, of course, the community area would represent an area with no native wildlife grazing. However, there is still low levels of livestock grazing. So to test these shifts in species composition between these areas with different grazing histories, we placed transects in these different areas, trying to pair them and minimizing any other environmental variables such as topography, slope, and aspect. So for instance, that would be a paired sample with all being in a sort of similar area. So the green lines are the transects, and then the red dots would be plots that we put out along these transects in order to measure the species composition. And even though we tried really hard to keep these transects straight, there are a lot of wild animals in the park, and although this one is straight for just display purposes, in reality this is what it looks like. because at some stage we were in a standoff between us, an elephant and lion, and in this case we chose life. So this one is not straight and a little bit zigzaggy, and although there are no big animals outside in the community area, there are these dense euphorbia stands that we just could not get through. So again, we chose life, and these transects are a little bit shorter than some of the other ones. Okay, so along these transects, they were 1 kilometer in length. We quantified the distribution of functional grass types, with lawn grasses being short stature, stoloniferous, and rhizomes, with bunch grasses being these tall grasses with vertical root structures. So at 10-meter points, we recorded whether the grasses were bunch or lawn, and we also then looked at tree density and herbivore presence by recording the number and identity of grazer dung piles as well as trails. And then for the species composition, we established these plots that were all on a north-sloping facing slope and at least 500 meters apart, at where we followed a 10-meter grid system where we identified the dominant grass species and recorded above-ground biomass using a DPN, which is that device over there, and looking at grass cover. So then just to analyze this and looking at the distribution of functional grass types, we did a generalized linear mixed effects model and included all of these variables that we measured, but adding a component for differences in the number of fires and for swale. Then to look at the community composition, first we started with a simple RDA, unconstrained, and this basically just uses the species data by itself so that we could see that if there are some patterns emerging between these different areas. And if there were, we did a constraint, which means that we're trying to see how much of the variables that we recorded are explaining this variance that we are seeing. And what we found is in the area with a lot more grazing, there was significantly more lawn grasses relative to the other areas. As you can see at the top here, there we go. There was slightly more lawn grasses in the community area, however, this was not significant. And then the variables that came out as important was number of trees, the number of fires, and this was over a 19-year period, and of course, herbivore presence, following the patterns that we would expect, meaning more trees, more fires, we would have less grass, lawn grasses, sorry, and with more herbivore presence, we would have more lawn grasses, of course. And then looking at the results from these community composition, again, the same pattern emerges with the area that has been grazed. It's over there in blue. Oops, sorry. Over there in blue, separating out relative to the other areas. Now, when you look at the constrained analysis that we did, again, the area with a lot more grazing separated out compared to the others. And when you looked at the species that were more closely associated with these different areas, you can see that all the species at the area with a lot of grazing were the lawn grasses, Panicum coloratum, Sporobolus euclatus, Euroclone mosambicensis, whereas those mostly closely associated with the other areas were the bunches, Panicum maximum, Themina triandra, and Panicum diastem. And in this analysis, again, fire came out as important, as did soil texture. So what does this mean? Basically, it means that long-term grazing by these mega mammals, as we expected, promotes the dominance of lawn grass species. And due to these functional differences between grass types, it suggests that the species dominance may influence important ecosystem processes, such as fire dynamics and below-ground biomass. So fire dynamics by maybe changing the fuel load, and below-ground biomass by accumulation of roots and root structures. So what's next? Now that we know that there are these differences in functional grass types between areas with different grazing histories, do these shifts in grass species dominance actually lead to a buildup of below-ground biomass? And what can we expect in terms of shifts in trait distributions? So using this data that we collected on the above-ground component, we identified the dominant species in the respective areas. Again, you can see the green is the lawn species, and the reddish is the bunch species. mostly lawn dominating in the area with a lot of grazing relative to the others. And then these species make up more than 75% of the dominance in these respective areas. And these I am then now targeting for excavation so that I can see what this actually means for the below ground biomass. So upon the recommendation of a paper by Ottaviano that I showed in the beginning, I am digging. I'm digging and looking at what's going on below ground. But no one told me there was going to be so much washing and sorting through organic material. So we're getting there, but it is a slow and painful process. And then, of course, because we're interested in these trait shifts, I'm also recording traits, looking at both above-ground traits and below-ground traits, so scanning roots so I can see what these differences are. Because we're interested in seeing how the traits differ within and between species, and depending on which disturbance they're exposed to, I will then next season, I will have these boxes which are called riser boxes, and they're essentially these transparent trays in which you can grow plants and trace the root structure. So this will just give a little bit of a more in-depth view of what's actually happening to these roots when exposed to different clipping treatments or fire. So that's sort of what I'm planning to do next, but at the moment we're still sorting through all of the roots, and I'm really grateful for the team that's helping me in the field because they're really keeping me afloat in the sea of roots that I'm now finding myself in. Thank you.
Thank you, Celeste. We do have a few minutes for questions, so if there's any questions for Celeste, let us know.
Thank you for the nice talk. I'm very happy that some people are also digging.
Yeah.
How deep are you digging, and are you doing these traits per species or for the community?
Yeah, so we're focusing on species level, and we're only digging down to 10 centimeters. At first, we were very ambitious, wanting to go down all the way to 1 meter, but that is a lot of soil. But we are also doing cores to go deeper down to 50 centimeters.
Thank you for interesting talk. I want to ask you whether you are aiming for rhizome biomass as well.
Sorry?
Rhizome biomass, whether you are planning to assess.
The rhizo boxes?
Rhizome biomass.
Rhizome bias.
No, biomass.
Biomass, oh, sorry. Rhizome biomass. Yeah. I haven't considered that yet, but I think that's a good idea. Yeah, for sure.
Okay, well, if there's any further questions, you can find Celeste at lunch. Thank you very much. Next up, we have Joris Kromse, who's going to be talking about some of the potential ecological cascading effects of the loss of white rhino. Thanks, Joris.
Thank you, Don. I've changed my title, just for the fun of it. So I'll be talking about how white rhino loss may affect climate warming, and I'm thanking Andrew Davies, I'm not sure if he's still in the room here, but for introducing this thermal landscape idea, and especially also the idea that animals themselves can actually shape this thermal landscape. So I will be talking about that. And I'm thanking all the co-authors that have been part of this effort, especially the first one, because I'm actually presenting the work of one of our ex-PhD students who graduated recently, Olli Hiverinen, and this is actually his work. So thank you, Olli. So animals can shape ecosystems. Many people have been saying that this morning, this whole week, in fact. So I'm just repeating this here. At significant scale sometimes. This is somewhere in the Eastern Cape. And what they can do is they can, for example, have potentially large positive but also negative impact on carbon stocks and carbon cycling. And that has also already been discussed a couple of times during this meeting. And because of that, and thank you, Duncan, for introducing the term rewilding a bit earlier today. Because of that, there's more and more people suggesting that animals should be part of the discussions around climate change mitigation. People have been talking about natural climate solutions, such as planting trees, People have also been saying that's often not a good idea. And people are now talking about alternative scenarios, and there's more and more talk about using animals and rewilding animals as part of a natural climate solution. For example, by then promoting soil carbon storage. What's often, so the big emphasis in this literature is actually on carbon. What's often ignored, I think what's actually almost always ignored, is that albedo is also a very strong driver of climate change. And albedo is basically the amount of energy, the amount of heat from the sun that is reflected back out of the atmosphere by the land surface. And I think all of you know albedo from wearing a black or a white t-shirt. So if you wear a black t-shirt, you're a little bit less happy if you stand in the middle of the sun than if you're wearing a white t-shirt, because the white t-shirt reflects the energy from the sun. That's basically albedo. So if you convert that to this landscape that I showed earlier, you have this white landscape, which is a white t-shirt in this case. It's a cooling landscape. It reflects more heat than this dark landscape. This is a difference. This is, as I said, a landscape in the Eastern Cape that actually has been induced by goat browsing. So on the right, you see a lot of speck boom, and on the left, all the goats have removed all the speck boom from this landscape. So a couple of years ago, I talked about how reindeer can do similar things up in the far north. And there has been actually quite a bit of work on how reindeer, by trampling and grazing on the tundra, can shift a shrub-dominated tundra into a grass-dominated steppe. And that this difference actually has a big impact on the albedo of these landscapes, where the albedo of the grass steppe is higher, so it reflects more light than this shrub-dominated tundra. Actually, Mariska has done some of this work, and the difference between these two states is a 2% difference in albedo. So this landscape here is 2%, has an albedo that's 2% higher than on this side. And this 2% actually makes a big difference. So it might seem like a very small difference, but a 1 to 2% difference in albedo between two different land covers actually can have a massive impact on your local climate. And there's actually a whole range of studies, but for example, this study discusses that very nicely, that the encroachment of woody species into grassy steppes, the albedo effect of that actually dominates the carbon sequestration by these woody shrubs. So the encroachment of woodies actually warms the northern parts of our planet. There's also this interesting work actually that looked at this same mechanism that I just discussed for the reindeer, but for these Pleistocene mammals that went extinct across the northern hemisphere. And they actually showed that the extinction of these mammoths and woolly rhinos can actually be linked to an increase of a warming event that occurred at that time across the northern hemisphere, especially the northern latitudes of the northern hemisphere. So what we were interested in is actually, can we then actually go from woolly rhino loss to white rhino loss? And do we see different processes happening with the ongoing white rhino poaching crisis through vegetation albedo feedbacks? So we know that white rhino can shape landscapes. They can have big impacts on all kinds of parts of the ecosystem. For example, they can shift these different vegetation states. They can shift lawns and bunch grass. They also have big influences on fire extent and fire frequency. And a lot of this has been shown before by several people also sitting in this room. What we then did, and this is something that I showed actually already 3 years ago, part of an initial field effort, is we looked at the albedo difference between open grassland and encroached grassland. So basically these two types versus that type. And we looked at the effect of burnt grassland versus unburned grassland. And we showed that the albedo difference is actually very similar as the difference between tundra, woody tundra and grassland steppe. So open grassland, this thing stopped working. Open grassland has about a 2% higher albedo than woody encroached grassland. And grassland that burnt recently has a 2% lower albedo than than the same grassland before the fire arrived. So woody encroachment and fire reduces grassland albedo by about 2%. So very similar to what the reindeer are doing up north. So what we hypothesized in this study is the following linkages between a white rhino loss, so losing white rhino from your landscape, and these ecosystem states. So the loss of white rhino would lead to an increase in grass biomass and a reduction of grazing lawn cover. It works, sometimes it doesn't. And secondly, the loss of white rhino leads to an increase of woody encroachment due to that direct impact on woody seedlings that are hidden within the grass. So really small seedlings that they would eat while eating bunch grass. So these are two direct impacts of white rhino on these ecosystem states. And then you have their indirect impacts. So because of reducing the amount of grass biomass and also of increasing grass biomass and reduced lawn cover, when you lose rhino, the effect of that is an increase in fire frequency and an increase in fire extent. And we hypothesized also an increase in cooler fires, because you basically get very frequent fires that lead to a reduced fuel buildup, which would still burn, but not as hot as a fuel that you leave for a couple of years. Well, that increase in more frequent and larger, cooler fires would then also induce woody encroachment because it removes the grass competition for the woodies. So that's the indirect effect of rhino on woody encroachment. Then this together then, an increase in woody encroachment and an increase in fires would reduce the albedo, as I showed earlier. Because woody encroached plots have a lower albedo, and burnt plots also have a lower albedo. So we set out to test this, and we used this, the same system as Celeste just introduced. So we looked at Gluclui in Flozy Park in KZN, and we looked at the whole park, and we divided the park in square kilometer cells. And we used data on rhino poaching for all of these cells to actually then look at the linkages between rhino loss and changes in vegetation, as well as fire and albedo. And so as I said, we looked at 1 kilometer grid cells. We looked at the 10-year period between 2010 and 2019. During that period, the park lost about 1,000 rhino. And if you look at the maximum here, so the maximum rhino a cell lost was 11 rhino. The minimum was 0 rhino. And then we excluded parts of the park that are really like forest or very close canopy woodlands, areas where we know that rhino have very limited impact on the vegetation anyway. And then we linked a whole range of products. So we linked the rhino loss data per square kilometer to a range of MODIS products. So we had the annual rainfall, in fact, came from long-term data from rainfall stations in the park. But then we had data from the MODIS fire product and data from MODIS woody cover and albedo products. And we linked them on this 1 square kilometer resolution to really look at this association between rhino loss, vegetation, fire, and albedo. So one of the first things we found here is that yes, so the rhino loss was very clearly related to fire. So we saw a strong increase in the mean burnt area and also a strong increase in fire frequency along this rhino loss gradient. So there's almost a doubling of mean area burnt along this if you go from the extreme rhino poached area to the situations where no rhino were lost. And the same is happening with fire frequency, although we see an interesting interaction here, where in the high rainfall area, there was actually no effect of rhino loss on fire frequency, probably because rhino cannot influence grass biomass to such an extent that it influences fire frequency. So secondly, we also saw a stronger annual increase in woody cover with increasing rhino loss. So what we found here is that in the areas that lost a lot of rhino, the rate of woody increase was faster than in the areas that did not lose any rhino. Again, there was a rainfall interaction here, where that effect was most strong in the high rainfall areas. In the high rainfall. No, sorry, we saw a more limited effect in the low rainfall areas in blue here. But overall, there was this increase there was a faster increase in woody cover in areas that lost more rhino. Well, and then we actually built a structural equations model because we were interested in these direct and indirect effects of rhino that I showed earlier on albedo. And what you see here is that both rhino loss and rainfall have a clear effect, directly and indirectly, on woody cover, on burnt area, and fire frequency. And that we indeed found both. Sorry, this thing doesn't work. we both found evidence for the direct effects, so from rhino loss to woody cover, and then towards albedo, as well as the indirect effects via the fire regime. So indeed, we found that actually there is through this SEM, we found evidence for an effect of rhino loss on albedo, and that the areas that lost more rhino actually showed a stronger reduction in albedo. So albedo is declining faster. So remember, that's the black t-shirt. It's becoming blacker and blacker and blacker. The areas that are losing most rhino are getting black more rapidly. So concluding, rhino loss seems to be associated, although this is a correlational study, so we still have to be careful. Rhino loss is associated with reduced albedo, which may increase local warming, larger burnt areas, and higher fire frequency in areas that lost more rhino. We also found this increased woody encroachment in areas that lost more rhino. This was associated with a stronger reduction in albedo of these areas. And interestingly, in the most extreme situations, so the areas that lost 5 or more rhino had an albedo reduction of about 1%. So that's actually along the same order of magnitude to the reindeer effect up in the far north. And as I said, a 1% change is actually a pretty significant change from an albedo point of view. So some of our next steps now actually include integrating the albedo effect with the carbon sequestering effect of the woody plants, because of course the woody encroachment also leads to a carbon buildup. So at the moment we're actually integrating the carbon and the albedo effects into one estimate. And then from that we want to estimate the net warming effect at the scale of the reserve. So what is the effect of white rhino loss at the scale of HRP in terms of its warming towards the future? Thank you. That was my talk.
Thanks, Joris. We have a few minutes for questions.
Carla.
Hi, yours. Thank you. That was great. The SEM that you showed, and it like flashed by very quickly, so I'm not sure that I interpreted it right, but it looked like you had rhino loss leading to an increase in fire frequency, which then had a positive effect on woody cover. Did I read that correctly? And if I did, what do you think is going on there?
Yeah, so indeed, so people think about fire in two different ways, of course, right? So more fire will reduce woody cover is often the immediate thought. But what we think is going on is that we are getting very frequent fires, so at least once every 2 years. So we think the fires, because of that, are actually cool fires that remove the grass layer, but do not actually affect the woodies very much. So speeding up woody encroachment, especially by actually promoting the growth of shrubs that are already there. So it's really kind of thickening. So that's what we are thinking. Of course, it's a correlational study, so I mean, there's no way of teasing things apart. But we indeed find this very interesting effect of more fires leading to more woody encroachment. More woody cover, I should say.
Yeah, thank you for that talk. I was just wondering, the loss of rhinos, how that compares to the biomass of all bulk grazers, because obviously there are multiple species that will impact the grazing lawns.
Well, in this system, if you talk about the whole grazer biomass, rhino are about 60% of grazer biomass, so it's a significant part of that. And there's another 20% which is made up by buffalo, which do not really use grazing lawns, for example. So, yeah.
Thanks, Joris. And if there's any further questions, you can catch them at lunch. But yeah, definitely a question that a lot of us are thinking about at the moment with the loss of rhinos. Next up, we have Amy Schroeder, who's going to be talking about the effects of clipping and invasion on African the African lawn and bunch grass communities.
Okay. Hi, everyone. Good afternoon. I am indeed Amy Schroeder, and I am a PhD student at the University of Bayreuth. I am supervised by Stephen Higgins, and before I start, I would just like to thank the funders, the institutes, and the projects for allowing me to conduct this research. So today I'll be explaining talking to you guys about some of my PhD work, where we looked at the effects of clipping and invasion on African savanna lawn and bunch grass communities. Okay. So the grass layer of African savannas, as we know, as we just heard from the two previous talks, can be described as a heterogeneous mosaic of lawn and bunch grass communities. So the lawn grasses, we know, can be described as being short, grow laterally, and are attractive to herbivores. And in contrast, the bunch grasses, they do grow tall, vertically, and not as attractive. So in an initial bunch dominant state, when there has been lawn invasion, the persistence of this lawn grasses is primarily dependent on frequent grazing. So if we just have a look at this small little picture over here, we just see that there's a bunch dominant state that's been invaded, coupled with some grazing. The little hippo over there. Anyways, so we know that it's possible for these grass communities to switch into one another depending on the feedback mechanism. And so this, of course, will change the structure and function of these grass communities from either a fire-driven system to a grazing system. Right. So Hempson and colleagues proposed a theoretical model showing that the grass layer of African savannas can be an example of an alternative ecosystem state. So here two states can exist where there's infrequent grazing and there'll be more bunch dominant state, as well as the lawn dominant state over there, which is seen with frequent grazing. However, for any of these switches to even occur, there are some requirements that need to be met. So here, an initial disturbance, such as a fire or an intense grazing event, needs to occur. And of course, a bunch dominant state can't just miraculously turn into lawn grasses. So there has to be some sort of lawn presence prior to this frequent grazing. Yeah. So of course, we see these beautiful mosaics in the field, and we can observe it with our eyes, but actually, empirical evidence trying to figure this out is challenging. So then we thought, okay, direct experimental evidence is lacking in the theory of alternative ecosystem states. So we were like, okay, what experiments can we conduct to demonstrate the theory? So if we take our example and we think of an alternative state model, we could assume that after an initial disturbance, lawn grasses will be able to invade a bunch grass community. And as we know, we need a positive feedback mechanism to create and maintain these systems. So in our case, we would have frequent intensive grazing. So we're checking boxes. And then what we were really interested in is investigating the grazing frequency and what this grazing, what grazing frequency would be able to maintain a dominant lawn system. So let's move on to the aims and questions. So we did a greenhouse experiment where we investigated whether lawn invasion coupled with grazing could persist and dominate a bunch grass system. And here we wanted to see how invasion of lawn grasses in a bunch dominant system may be influenced by these grazing frequencies. And here to simulate grazing, we use clipping. So we also wanted to see under which clipping frequencies are these lawn grasses actually able to invade. And then to interpret the results, we used the Bayesian state space model. And so I'll just get into it. So basically, to mimic a grass system, we grew one lawn grass species together with 3 bunch grasses. We had two mixtures. The lawn grasses that we used are common lawn grass species found in African savannas, as well as the bunch grasses. So we chose Cynodon dactylon and Penicillium cladistium for the lawn grasses, and I'm sure you're all familiar with those species at the bottom of the bunch grasses. So just for the purpose of this talk, I'll just be focusing on mixture one, because we did have similar results or similar patterns for both of these different mixtures. And yeah, so these species don't necessarily occur together in the field. However, we really were just interested in seeing if there's like a dominant, or if there could be a switch between lawn or bunch to lawn. Yeah. Okay. So then what did we do? So this was our experimental design, where we had 63 plants in each box. Here we had 5 boxes, mesocosms. Again with the mixture one over there. As you can see, the box over there, it's very big. So we planted our bunch grasses randomly on a grid, and we put the lawn grasses systematically around or between the bunch grasses. We did this because we wanted to have the ratio of a bunch or a lawn grass within a bunch grass in the box equally. And then, so we used the substrate perlite. It's a light substrate, and it was really helpful because our boxes are huge, and it would have been very heavy with sand. And then we watered each box once a week until saturated, and we gave them all the same amount of nutrients. So then we had to move on to our clipping regime. So before we even thought about our clipping regime, we had to initiate an initial disturbance. So after 4 weeks of growth, all the boxes were clipped, and this represented a disturbance, a very intense disturbance. But then for our clipping regime, we had frequent to infrequent, and for example, every week this pink, I don't know if it's pink, but pink box got clipped every week, and every 2 weeks the yellow box, so each box was subjected to a clipping treatment. And then the most infrequent was clipped every 6 weeks. So here we just see a picture of regrowth after being clipped every 6 weeks or 2 weeks. And then the experiment lasted for 16 weeks. And because we couldn't get any large mammals to come and graze our boxes in Bavaria, so we just used a normal garden scissors. And we clipped all the biomass, we dried and weighed them, and to quantify the dominance in each box, we used the biomass measurements. So then we used, to interpret the results, as I said, we used a state-space model. So here we developed a plant growth model in a Bayesian framework. So just one thing about it, it's a really powerful tool to actually observe hidden processes given your observed data. So here this model is actually composed of it has 2 compositions, and here we have the observational model, and as you can see here, each state is actually independent from one another, and in this model is where the observed data that we had from our experiment went in. So in our example, we actually, when we clipped, for example, every 6 weeks, we only have a biomass data for that clipping event. So what happened in between, we don't know. So that's where the process model comes in, where the process model, it influences the observational model, as well as every time step in the process model is actually dependent on the previous time step. So what happens in this process model, it's not as scary as you think. So because our state variable is biomass, and that's what we want to predict, we had to estimate some parameters that actually affect biomass. So here we estimated growth rates as well as clipping rates and events, and this was basically trying to mimic our experiment. So here the clipping rate, for example, we fixed that at 90%, so that basically means that when there's a clipping event in our model, it means that 90% of the biomass is taken away, and this is what we also try to do in our experiment. And then we use the Laplace's Demon package in R, and to explore the posteriors, we use the Markov chain Monte Carlo method. Yes. Okay, and then our experimental data. So this is basically just showing total biomass after the end of the experiment, so our end harvest, and on the x-axis we have the clipping regime, so frequent to infrequent. So here we see that when clipping is actually frequent, we see that lawn grasses are way more present in the system, and that's what we saw from Celeste, so in the field, which is woo. And then, and then as the clipping becomes more and more infrequent, we have bunch grasses. So this is pretty cool just to see from our data. And then, so then we had to, of course, do some checks to see if our model actually worked, and it did. So it was flexible enough to replicate the empirical estimated grass biomass, as well as describe the observed patterns from the experiment. So that was pretty cool. It took a while to get here, but we made it. And then just to talk about some of the estimated parameters, so here we have growth rate. And of course, we could estimate a growth rate for each species, for each clipping frequency. And of course, growth rate, I mean, we can get a lot of information about biomass from this, but a low growth rate doesn't necessarily mean a very low biomass. But here we see in A and B, so A is basically most frequently clipped, and B every 6 weeks that it was clipped. We see that cynodon, which is represented in blue, which is the lawn grass, actually has a high growth rate, and as the time goes on, the growth rate actually decreases, and the same for all the other species. And in B, we see Eragrostis over there having a high growth rate, and again, as time goes on. And this could be, something has to affect growth rate here, so it could be the clipping, the constant clipping. And the growth rate here was the proportion of biomass increased in each time step. Okay, so then if we look at the predicted biomass, here again we just have the predicted biomass percentage per week. I do have the original data if any of you would like to see it. It's just visually this is a better way to show it, to show the big differences. But here we have time on the x-axis, and basically we can see that the model or the predicted biomass over here, we see that cynodon, which is the lawn grass, again is dominant in this box throughout the clipping or throughout the time steps. Here we see Eragrostis, which is the green, which is a bunch grass. It's present. All the bunch grasses are actually present and did not get kicked out of the system, but they're present, but you can see that it's quite a low percentage. And this could be attributed, of course, to cynodon, a lawn grass, having mechanisms to actually deal with being defoliated all the time. And for example, Eragrostis not so much. And then if we look at B, we see that, in B we see that the cynodon is actually has a low percentage over time, but again, not being pushed out of the system, and Eragrostis is pretty strong. And here, this could be attributed again to these bunch grasses actually growing tall and shading out the lawn grass, so it's not able to actually have a chance. And of course, we know light is one of the most important factors in lawn persistence. So that's pretty cool, I think, that we could see. And then, so if we bring it back to the theory of alternative ecosystem states, we can say that, there's a strong feedback for lawn grasses to actually persist, and invade successfully when clipping or grazing is frequent and intensive. Whereas it's a weak feedback when it's most infrequent. So that's, I think it's pretty cool to see that. And so then on to conclusions. So basically in our study, we were able to show that frequent intensive grazing together with the lawn grass invasion shows that the lawn grasses are able to persist. And as we know, herbivores are big factor that shape the landscapes in African savannas. So here we could show that under frequent grazing, these grasses can be very dynamic, or these grass communities can be dynamic. And then, of course, understanding the underlying processes that actually initiate these changes are valuable, especially for management. As Celeste and Yurich was saying, it can change the function and the structure from grazing to a fire system. And we need to be careful. And then also greenhouse experiments and Bayesian state space models are useful for developing models of plant community dynamics. I know a lot of people are scared of it, but you don't have to be scared of modeling. And yeah, so I think this is actually a really good tool, especially when trying to provide some information about theories that are very difficult to actually observe in the field. So with that, I'd like to thank you all so much for your time.
Thank you, Amy. You've used your time well, so any questions we'll have to wait till lunch, but thank you very much. Really interesting work. Next up, we're going to hear a little bit more about the long-term herbivore exclosure experiments here in Kruger. Helge van Koller is going to talk to us about some of the top-down control of herbaceous communities from these exclosures. Thank you.
There we go. Hi, everyone. So we know by now that various abiotic and biotic forces interact to shape and drive plant community structure and ecosystem functions in savanna ecosystems worldwide. And the main top-down drivers of herbaceous vegetation dynamics are herbivory, fire, and rainfall. However, the functioning of these various mechanisms are increasingly being altered and weakened in human-impacted savannas. And this is through the loss of diverse suites of native herbivores with which these systems evolved, eliminating them from the system completely or replacing them with domestic livestock. Other forces include modifications to natural fire regimes and increased frequencies of extreme droughts and rainfall variability due to global climate change. And these alternations will inherently impact the ecosystem function and services provided by savannas globally. So we know that ungulate herbivore impacts on savanna plant communities have been well documented. However, less attention has been given to the effect of different sized herbivores. And these impacts may differ between species in terms of various aspects such as body size, feeding mode, forage behavior, and nutritional requirements. And although studies have begun to address the taxonomic and functional identity of forbs and grasses, their response to either the total or partial release from herbivory remains less explored, especially the forb component. But we do know that herbivore release from disturbance-maintained ecosystems causes shifts in herbaceous life-form dominance. And this is through competitive exclusion effects via the accumulation of biomass, moribund material, and limited resource availability. And then furthermore, we know that differential consumer pressure and also the duration of disturbance impacts may have heterogeneous patterns on top-down control of community dynamics. So to test for the effects of different sized herbivores on herbaceous species composition, we used long-term data that was collected in an exclusion experiment that was established in November of 2006. And here we focused on the triannually burn treatment of the experimental burn plots that was established in 1954 near the Satara rest camp. And the experimental design consisted of three treatments, namely the full exclosures, where all herbivores were excluded, and then the partial exclosures, which excluded all animals with a shoulder height greater or equal to 0.85 meters, for example, zebra, wildebeest, and buffalo. But these exclosures allowed for small and medium herbivores such as impala and steenbok, and then also the open access areas. So these exclosures provided a gradient in herbivory ranging from no herbivores to medium and small herbivores only, and then the open areas with the medium and small herbivores plus large herbivores. So the Central Kruger National Park supports a wide variety of mammalian herbivores, with impala, zebra, elephant, and buffalo being abundant. And these triennial burns generally also attract a wide range of grazers and browsers, potentially due to the provisioning of periodic high-quality forage and also a variety of forage resources such as forbs, grasses, and woody browse. So the Satara region receives a mean annual rainfall of 550 millimeters, with 80 to 90% of this falling in November to March. And to determine the effect of duration of disturbance on herbaceous species composition, we focused our analyses on 3 sampling years, namely 2007, which we called the early exclusion, which was 1 year after exclosure establishment, and this year received a below average rainfall of about 108 millimeters. Then 2013, which we called the mid-exclusion period, and here was an above average rainfall of about 210 millimeters, and then the late exclusion, which was 16 years after exclosure establishment, and that year received an above average rainfall of about 90 millimeters. Okay, so the varying rainfall quantities for the sampling years allowed us to link herbaceous species assemblages to rainfall variability, because we know that interannual rainfall variation is the most important driver of herbaceous layer dynamics in semi-arid savannas. And we also found in another study with a granitic, in a granitic study site with a slightly higher rainfall that rainfall tended to outcompete the effects of herbivores. So we wanted to see if it was going to be similar for this study site. So at a coarse scale, when including all herbivore treatments, the full, open, and partial, and all of the exclusion duration time periods, we can already see some clustering, some clear clustering in a 3-dimensional space. However, this clustering was not in terms of herbivory, but rather in terms of duration of exclusion. And as you can see, the mid-exclusion time period showed the strongest response in herbaceous community, with the early and the late exclusion time periods clustering closer together. So to tease out whether some of the herbivore effects got obscured in the NMDS, we completed a canonical analysis of principal coordinates. And as you can see, it's still, the composition still split up mainly in terms of the exclusion duration periods. However, we did see some effects in the late exclusion time period, where the late open differed significantly from the late full and the late partial. And this was, just to remind you, 16 years after the exclosures were established. And these changes were mainly driven by grasses, supporting the findings of Bergpile and colleagues, who reported significant increases in grass cover with herbivore removal in the same study site. And moreover, we also know that long-term herbivore exclusion has been known to favor palatable perennial grasses. The low forb cover in the late open treatment was, however, a bit surprising, because we would expect that the increased open spaces and the decreased competitive exclusion would have favored some forb species. However, the rainfall was also above average for 2022, which might have caused these forbs to be outcompeted for resources such as water, nutrients, and light. So the overall cover was mainly dominated by grasses, irrespective of herbivore treatment or exclusion duration. And this reinforces the fact that indigenous African grasses are resilient to various savanna disturbances. However, I would like to focus your attention on increased forb cover in 2013, which may be ascribed to the above average rainfall of about 210 millimeters in this sampling year, and forbs also having the ability to make better use of improved conditions. And these patterns can furthermore be attributed to post-drought dominance of the annual forbs. So, as we can see by now, that the herbaceous assemblages grouped by duration of exclusion rather than by herbivory, and when considering the specific species that contributed to these differences, and also the dominant ones at the different duration of exclusion time periods, it seems that rainfall best explain these herbaceous assemblage responses. So for the mid-exclusion time period in 2013, with that very high above average rainfall, we saw that 67% of the dominant species were forbs. And like I mentioned, forbs have the ability to make better use of better environmental conditions, such as increased water availability. And half of these forbs were palatable annual forbs, which also form a major part of soil seed banks, and they have the ability to emerge quickly when conditions become favorable. For example, above average rainfall after a drought year. And then forbs contributed most to the differences between the different sampling years. So the presence of palatable, perennial, and annual forbs reinforces the importance of us to include this life form in savanna ecosystem studies. As would be expected, late exclusion was dominated by palatable perennial grasses, attributed to the combined effects of herbivore removal and above average rainfall. And below average rainfall was recorded for the early exclusion year in 2007, when the exclosures were only 1 year old. And they were also dominated by palatable perennial grasses and forbs, suggesting that this system is resilient and able to provide important ecosystem functions and services when conditions aren't favorable. To zoom in a little bit, so at a finer scale, when I separated the different herbivore treatments, you can see that the patterns were mainly similar across all of the treatments, suggesting that something other than herbivory drove these patterns, because it didn't really matter which herbivores were present or excluded, the patterns remained the same. And we then saw that it was the mid-exclusion time period that was clustering more towards its own every time, suggesting once again that rainfall may have outweighed the effects of herbivory here. So to conclude, contrary to what Birkpile found, although they only focused on species richness and diversity with herbivore removal, they didn't find any real effect of herbivore removal on the triennial burns. We found that herbaceous species composition was significantly affected by herbivore exclusion. However, this was only evident for the late exclusion time period, so at least 16 years after the exclosures were established. So long-term monitoring in the long term, it seems that herbivory increases in importance, whereas in the short term, rainfall seems to be a bigger driver. And then long-term monitoring is therefore crucial for us to understand the effects of disturbances in savanna ecosystems. And then we found that differential consumer pressure did not result in heterogeneous patterns in top-down control, because all of the response patterns were similar across all of the herbivore treatments. And then for this system, forbs were mainly responsible for differences in herbaceous species community when rainfall was above average, reinforcing the importance of us to include this life form both at a taxonomic and a functional trait level when trying to understand savanna systems. And then lastly, rainfall variability best explained patterns in herbaceous species assemblage response, and therefore we can conclude that interannual rainfall variability is considered the strongest driver of herbaceous layer dynamics in this system, as it outweighed the effects of herbivores. Thank you very much.
Thank you, Helga. I think we have time for one quick question, if there's one.
Very nice talk.
Thank you, Kevin.
I'm just curious, have you looked at any of the other years of data to kind of try to parse out total rainfall with some of the patterns?
That's one of the next steps, just to test the robustness of these results. We're going to see if the patterns were similar in some of the other sampling years. So yes, that's part of the next step, so I don't have an answer just yet.
Cool.
Thank you, Kevin.
Thank you very much. We're moving on to the last speaker for the session, who's our recent award winner, Francis Siebert, who's going to be talking about some of the functional differences in grasslands and treeless savannas. Thanks very much.
Thank you very much, Don. Good afternoon, everyone. Yes, today I want to throw out this daring question of, can we consider a treeless savanna a grassland? Just to put everyone into context, specifically for the new visitors to the savanna of South Africa. You are situated in the savanna biome of South Africa, where there's a considerable cover of shrubs and trees in the landscape and a continuous layer of herbaceous vegetation. But imagine yourself within the same biome in Kruger with open vast landscapes of just grass everywhere, a little bit forbs in between, of course, and then only scattered trees. Would you consider this as a grassland? And is it important to know whether it is a grassland or savanna? So merely about 80 or 100 kilometers from here, up in the escarpment, higher altitudes and also higher rainfall, we get these really open landscapes covered also by grasses, maybe some scattered trees as well. So physiognomically, structurally, these systems look very similar. But do they compare floristically, and do they compare functionally? And does it really matter? Well, in the South African context, I will try to provide you a little bit more evidence that they are different. But I would really like also some insights from other people also working in subtropical and tropical savannas and grasslands, whether you have observed similar patterns. So my presentation will include 3 different questions. Of course, I will start with the floristics, only the herbaceous layer, looking if these treeless savannas and grasslands differ floristically. Then also attempt to look at some of the functional diversity. How does it compare, and is it different at all? And then lastly, so what? Is it important that we know this? So, thanks to Wynand, one of my students, he created this map for me. So my husband and I, Stefan Siebert, we were sampling savannas and grasslands for many years, actually as a hobby. Did 10 by 10 square meter plots because he also has a similar passion for the forbs than I have. And so we accumulated a database quite extensively on these grasslands and savannas in South Africa. And we decided to extract all the data of the savannas with a tree cover of less than 5% to give us an idea of what some people would call a grassland can really be compared to the grasslands, what we know in South Africa. So these data also, these plots is also in the different rainfall regions of South Africa, covering some mesic savannas and also mesic grasslands, and also on the arid spectrum. So in these 10 by 10 meter plots. We did full floristic surveys, grasses and forbs, of course. But we extracted only the dominant herbaceous species in these plots, and also gave an index value that not only included the frequency, but also the abundance of the species to get a better feel for the dominance, because that's what mostly shifts the difference between these systems. So the first question, all right, clearly there's a quite distinct difference in the dominant species in grasslands and savannas in the South African context. I just need to remind you that. We split grasses from forbs. We actually expected a stronger overlap, but still there was a clear differentiation between grasses in these systems. Forbs as well. Then we looked at the dominant families in these different ecosystems that, well, they are equally ancient. They have also all evolved with herbivory and fire. But there's a quite strong dominance by Aceraceae in the grasslands and Fabaceae. And in the treeless savannas, quite a distinct number of different families, of which the more important ones are the Acanthaceae and the Maranthecae. But I also want to point out the importance of Fabaceae in this system. So I'm not only biased towards forbs, and I even have a picture for all my grass colleagues. And if you look closely, you can see some grasses there in the background in our forblands. So getting back to this, later on I would like to illustrate the importance of these two families in the grassland ecosystems, because I don't know, I haven't seen all the grassland ecosystems in tropical savannas, but usually Aceraceae daisies are being associated with being weedy, ruderal species. But later on I will show you some amazing examples of these plant families in our grasslands. So getting back to my first question, floristically, I think it's quite clear that there's a difference between South African grasslands and savannas. Now getting onto the functional component, it was of course a little bit harder, but we expected a much stronger overlap in the functional composition in these two ecosystems, because they have similar drivers and they are equally ancient, and they are also both, by the way, threatened by tree planting initiatives. So again, this is familiar to you, a very nice framework by Sally, Gareth, and Caroline. But before I get to that, we just simply looked at the annual and perennial separation of plants in these two ecosystems. In the grasslands, it's perennial dominated, very distinctively perennial dominated. Those of you who work in the grasslands in South Africa would know that once there's a destructive disturbance, there are no native annual species that colonize that area. Most of the time it is alien invasive annual species that come in. So there's really a very small proportion of annuals in the grasslands, opposed to the savannas, the treeless savannas, but they are still perennial dominated, as was also illustrated by Helga in her previous talk. So the next step was to put all of this into plant functional response group. Now, the plant functional response group was born from, of course, after discussions with Gareth and Nikki through the BII project, where we also tried to do this for forbs, having these categories of avoiders, resistors, and tolerators. So during the Biodiversity Intactness Index for Africa project, led by Hayley Clements, to assess the biodiversity of such a hyper-diverse continent in terms of plants, we had to put them in particular plant functional response group if we want to see how they would respond to disturbance. And that was actually a very unique opportunity for me then to test these plant functional groups to the data that we have to differentiate between grasslands and savannas. So this is, unfortunately, I don't have time to go into detail about how we got to these different plant response groups. For forbs, yeah, we also have forb lawns, fire resistors, herbivory avoiders, etc. So if we take the floristic data set and we apply these groups to the grasslands and savanna, what do we get? Right, there are some overlap, but still quite a distinct difference between the grasslands and the savannas. When I separated the grasses and the forbs, there were quite an overlap with grasses, and it's of course because both of these systems have evolved with herbivory and fire, and during a defoliation event of fire or herbivory, they can tolerate these conditions, so they can be considered as generalist tolerators. But on the grassland side, according to the SIMPA analysis, the plant groups that were most responsible for the split was the dense tufted, the fire resistor. So we can postulate that for grasses, although there's an overlap, there's a strong split towards fire-adapted grasses in the grassland biome. For the treeless savanna, it was mainly because of the sparse annuals, and I did remind you that that's because of the lack of annuals, or the very poor abundance of annuals in the grassland biome. Right, so for the forbs, we've seen similar split with the generalist tolerators. There are many, and that also makes up the largest component of the forbs in both of these ecosystems are the ones that can, during the event of defoliation, they have the ability to resprout. And that's what we are also looking into with some of the other projects that we are busy with. But for the forbs, the functional groups, more towards the grassland side, the generalist tolerators, but with underground storage organs, and also other fire tolerators, meaning that they are either toxic and they can avoid herbivory. And on the treeless savanna side, we have seen a lot of weedy and palatable annuals because of the split, because of the lack of annuals in the grassland, but also palatable annuals and grazing tolerators and grazing resistors. So in terms of functionally, I think, although of course we need to still do some proper trait measures to test this properly. But then it comes to my last question. Okay, so what? And I think this is more of an advertisement for grasslands in South Africa. Because they are hyper-diverse, they are so poorly conserved, they have so many ecosystems functions that they provide to us. So calling the treeless savannas a grassland does not really acknowledge the value of our grasslands. So coming back to the real grasslands in South Africa, with a lot of inspiration from my Brazilian friends and colleagues, of course we started digging, and this is what you find in the grassland, and this is an illustration of the dominance of the Asteraceae in the grassland ecosystems. They are not annuals, they are driven by these perennial, very carbon important functional groups in these systems. And this was also introduced by Mariska in her talk, that we are going to do some more digging and some more exciting stuff to just to promote this. And this is, of course, the Fabaceae. So if we refer back to Kevin's talk about the drought responses here in the treeless savannas. They were dominated by annual Fabaceae after the drought, whereas this is highly unlikely to find in the grassland biome. And this is some of the Rubiaceae, also a dominant family in grasslands. Right, so in conclusions, I would like to test the idea of savannas in terms of the South African context. They are very important to support our megafauna. They have herbivore-adapted traits and tolerance. But what about the grasslands? They're not really big five habitat. We do have our megafauna in the grasslands, and of course eland that only forage on forbs. So this is a term that was also suggested by Joris, so I want to give credit to him, but I think grasslands rather support mega forbs and not as many mega herbivores. Thank you so much.
Thank you, Francis. That does wrap up our session, so if there are any questions, perhaps you can just catch her at lunch. Before we do head off, I just want to say thank you to all the speakers for the session. It was a really fascinating set of talks. And just one quick announcement to all speakers for this year's meeting. If at the end of this, the talks will be uploaded to the SAMParks website. If you would prefer your talk not to be uploaded, please just find Jackie and let her know, and then the talk won't be uploaded. But otherwise, thanks very much. That wraps up the plant herbivore interaction session. I'm sure if you want to see more, you can head to the center island in the luncheon area. Thanks, everybody.